on behalf of and myself to constant international work on development of animal models for metabolic disease. Metabolic diseases, one of the major metabolic diseases is diabetes, the non-communicable disease, whose prevalence in the member countries of the OECD is significantly high. Therefore, comstech has invited now cited from Bangladesh, Professor Dr. who's affiliated with the Department of Biochemistry and Molecular Biology, Dhaka International University, to conduct this workshop on metabolic diseases, focusing on diabetes. He has 2 decades of research work exclusively on diabetes. This workshop will highlight and discuss development of animal models, which are used in the study of the metabolic disease. And these animal models play an extremely important role for the elucidation of complex biological mechanisms involved in disease development and to expedite drug discovery process. To begin our workshop, we will begin our workshop with the recitation from Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الرحمن علم القرآن خلق الإنسان علمه البيان الشمس والقمر بحسبان والنجم والشجر يسجدان والسماء رفعها ووضع الميزان ألا تطغوا في الميزان وأقيموا الوزن بالقسط ولا تخسروا الميزان صدق الله العظيم.
I welcome our guest, esteemed guest from Kazakhstan, Dr. Selik, to our international workshop. With this, I would request His Excellency Professor Dr. Muhammad Iqbal Chaudhry, who is the Director of ICCS, as International Professor and an expert chairholder on medicinal and bioorganic natural product chemistry and coordinator general of comstech, for his welcome remarks.
Thank you, Khazima. Auzubillahi minash shaitanir rajeem. Bismillahir rahmanir rahim. Excellency, my dear brother, former senator, advisor to the president, and also the head of National Audit Committee of Kazakhstan, Professor Dr. Serik. My younger brother on my left side, Professor Dr. Muhammad Hafiz, Hafiz Rahman, for being with us, and all of you from various universities of the country and also from OIC member states. You see, this event is a very serious event in the context of some serious challenges which we all face. Think of the world today. There are several myths which we have. For instance, we have the myth that average life expectancy is increasing. The fact is that we do really live longer, but we live longer with diseases. So we live a miserable long life. So if you think that you are living longer, then think of it that you're actually living with many chronic diseases, which eventually lead to a serious compromise of the quality of life. So this myth that we are increasing the lifespan is certainly is more of an illusion, because this is not the quality of life or the life which we desire to have. It is more of a life in which we suffer, and suffer even longer than our ancestors used to live. Second myth is that the pharmaceutical industries, which are designed to develop new medicines for treatment of human sufferings, do that with the intention of helping people. This is the biggest myth and perhaps a major joke, because pharmaceutical industries are businesses. They work on the model of feasibility. Their decisions to develop drugs are based on economic feasibility, that how much you would invest and how much would you eventually earn in the long term. And therefore, if we expect them to invest into prevailing and poor man diseases, this is again an expectation which is certainly not an expectation which will ever be fulfilled, because pharmaceutical business is a business like any other business, which is designed to make money and not to help the humanity at large. So you hardly find any pharmaceutical company working on poor man diseases or diseases of a chronic disorder. There is a myth also that the diseases can be cured. The fact is that majority of the diseases cannot be cured. The fact is that majority of the diseases are just only managed, because you think of metabolic disorders, you think of arthritis, you think of cardiovascular diseases, you think of dementia, pain, arthritis, and numerous other diseases. You don't really have a treatment, you just have a management. So the entire paradigm of drug discovery and development is based on a myth which has been successfully created as if that they are finding treatment. In fact, a major survey was conducted in North America in 36 big hospitals, where people were asked when they are entered into tertiary care for serious chronic conditions, what they eventually learn. And out of patients, survey said that they learn to live with the disease. That's the only thing which they learn from when they come out of hospital. So the diseases are not treated, they're only managed. And they're managed not by design, not by default, they're managed by design, because if people live longer and the diseases are only managed, then they become a constant source of income. So don't even think that any disease is currently meant to be treated. It's only meant to be managed, because you become a chronic source of constant source of income. Then there is a myth that one single system of medicine can treat all human sufferings. Certainly not. The fact is that our ancestors used to live often better life than us. Tell us one important thing, that they knew how to treat diseases and they knew how to prevent diseases. They also knew to live a productive, engaged, and healthy and happy life than us. So, and they were not using Western medicines. So I remember that I was traveling in Sindh, and then I found someone who was traveling with me in their jeep. Please do come. And he told me his age, which I could not really believe. And then he said, I have never used any Western medicine, that I manage all of my ailments and sufferings by using traditional medicine. So not a single system of medicine capable of alleviating and treating all the diseases. And therefore, human wisdom in terms of its complexity and antiquity is required in a holistic, integrated manner, so people can benefit from the experiences of humanity at every corner of the world for the treatment of diseases, whether they're herbs, meditation, yoga, you just name it, intermittent fasting. All of this has to be integrated into a larger medical system. And perhaps the most important for us to start developing our own drugs. If we expect that pharmaceutical company at one day would have a change of heart and will start investing heavily into the treatment of our diseases such as malaria, schistosomiasis, leprosy, dengue, and others, is just a myth again. So we don't really expect them to invest into our diseases, and we need to do it by ourselves. And for that, we need to develop our own science. And for that, we don't really need to follow the US FDA model, which requires $2 billion US, which requires 10 to 15,000 scientists working together, which requires 10 to 15 years for development of a single molecular entity. Certainly, in kind of an input which we can never provide. So we need to develop our own paradigm for drug discovery and development. That is where the role of Hafiz comes, because there we need to have our own science. Now, in order to develop our own drugs, of course, we have a very large knowledge base that we have many of these traditional remedies available for us. And but all traditional remedies need to be scientifically validated. They need to be validated in terms of their safety, efficacy, consistency, standardization, and a number of other things. How would you validate their efficacy in disease model? For that, you can't really try that directly on human beings, though they are pharmacopoeial drug. Any laboratory discovery, the work which you conduct in your lab has to be translated into human use, and that you can only do by animal model. Therefore, animal models play an extremely important role, because animals mimic many of the biological and pathological conditions of humanity. Therefore, they are the best option, perhaps the only option of conducting a translational research. Hafiz has done a phenomenal work on a very important aspect of metabolic disorder. There are about 600 of metabolic disorder. And diabetes is certainly the most important one, which is linked with lots of other diseases such as cardiovascular, CNS, and others. Hafiz has worked on a very important aspect, which is called pre-diabetic model. You see, every diabetic person go through a very long period of about 10 to 15 years before he or she become fully diabetic. It's called pre-diabetic or impaired glucose tolerance and other, plenty of them. There is no science available on it. Nobody wants to work on it, because if you can treat IGT pre-diabetic, then you would not have a diabetes. And if you don't have a diabetes, you would not have a money for the lifelong use of anti-diabetic medicine. Dr. Hafiz has worked on it and successfully developed some very interesting models for pre-diabetic conditions in animals, generally rodents. Dr. Hafiz is a very a prominent, extremely talented, very hardworking researcher. He completed his PhD from University of Kumamoto in Japan in 2005, and soon after he joined us. So I searched him from a very long list of CVs, and then I found him. I said, this is a person I would like to bring to Pakistan. So he has a dual appointment. He's a professor here in Karachi at the International Center for Chemical and Biological Sciences, training large number of people. And of course, because of his own personal reasons, he has to stay in Dhaka, and he's a professor in Bangladesh University, International University, Dhaka International University also. Hafiz Rahman has many, many good publications, and certainly his presence for here in Comstech is an extremely important event. We have very large number of people who can benefit from his experience. Hafiz, thank you very much.
Thank you very much, sir. Ladies and gentlemen, our speaker has more than 60 international publications to his credit. He is also a recipient of Mixed Fellowship from Japan, and he has won several Mixed grants. Dr. Hafiz currently is the professor and chairman of the Department of Biochemistry and Molecular Biology from Dhaka at Dhaka International University. With this, I would request our guest speaker today, Dr. Hafiz Rahman, for his remarks, please.
Thank you, Kajima, for your nice introduction. So, ladies and gentlemen, those are physically present, or those are in voice countries in online. Assalamu alaikum to everybody, and very good morning. So, thank you very much, Comstech, to arrange the workshop, and thank you, Dr. Professor Iqbal Chaudhary. Nice word, kind words for me as well as for this important disease. It's my immense pleasure to be present here and will be conduct this workshop. This is timely being workshop, very important and timely needed. So as Dr. Iqbal already mentioned, so disease. So everybody nowadays, we are living long with some diseases. Especially after 40s, for whole life, we are suffering from disease. And due to some most urbanization, modernization, Coca-Colaization, McDonaldization, so our food habit also will big changes. Your traditional system to Western system. So in this ratio nowadays, all over the world, so the obesity, overweight, dyslipidemia are increasing. So in one of every 3rd people is suffering from obese, overweight, and 1 out of 10 suffering from obesity. And dyslipidemia also increasing rapidly all over the world. So diabetes is a major metabolic disease. So it is maybe it's cause of many diseases, or it's a result of many complications. So drug is designed and developed by the Western countries. This is designed and developed for their needs, not for ours. Initially, this is designed, cure, or management for their peoples. However, the same drug may or may not be correct, or may not be effective, safe, effective for all over the world. So the difference is the human subjects, Asian subject, Western subject are different. So human physiology, though it is similar, it is not same, but similar. So WHO mentioned that the criteria for the obesity for Western people, it is more than 25. But the obesity for Asian countries, it is more than 20, it is 23. So there is a difference. So Asian people are mostly non-obese. So however, the medicine is the same. Patients are different. Medicine is the same. So in case of diabetes, so there is type 1 diabetes and type 2 diabetes. So in case of type 2 diabetes, we have some classical type 1 diabetes, not type in Asian people, in India, Bangladesh, Pakistan, we have some disease, some diabetes. They're not either type 1, not type 2. However, that treatment is going to the classical system. So therefore, we need our own, we need our own model. We need our own medicine for the betterment of this, for human beings. As comstech is taking some initiative, this one. So for this development of any drug, we have this drug or traditional medicine, you have to gather, you have to pass through some preclinical studies. All the preclinical studies should be done in the animal model. Then we call it normally a small animal model. So I have a long experience in diabetes research, especially in Bardem, Dhaka, and finally in Dr. Panjani Center, ICCBS University of Karachi. So in this workshop, I can share my experience and expertise, and hopefully the participant will learn more how to handle animal, how to learn the human animal behavior, how to develop good model, how to test your compound for the better development of drug. So one thing I mentioned here that, so sometimes money is a matter, but money is not solve the problem all. During COVID, it's a good lesson. So disease is there, COVID is there. We have all people's, all countries money, but it is medicine is not available or vaccine is not available. So it's priority based. So this is the time to think and rethink for our own disease, own disease and own medicine. So the mission and vision of comstech for the health, medicine, science, technology, and higher education. I think this workshop will cover all of the aspects of science, technology, drug discovery, as well as model development. So with these few words, again, I thank you, comstech, for arranging this workshop. And Dr. Especially, I'm grateful to Dr. Iqbal Choudhury to inviting me in this international workshop. It's my immense pleasure to be here. And hopefully the participants from home and abroad, all over the YC countries, will get some idea for your science, biomedical research, and drug discovery. So next 3 days, I'll be with you and share my experience and expertise. Hope you will enjoy my workshop. Thank you very much. Thank you for it.
Thank you very much, sir. With this, I would request His Excellency Professor Dr. Muhammad Iqbal Choudhury to give detailed today to our guest speaker, who is also comstech distinguished scholar.
Why don't you come and join us? And all other foreign guests?
All the foreign participants, kindly come. Dr. Munir,
From Afghanistan.
Sweden, Uganda, Yemen.
We'll turn like this. We'll have one place. Okay, so
picture इधर यहाँ
next 5 minutes.
Ladies and gentlemen, I would like to thank our guest speaker, our participants who are present here in person, and virtual participants for their time. We can now have them working at tea break for 20 minutes, and we will be back here by 10:50. Please serve outside in their dressing room. Thank you very much, ladies and gentlemen.
This is okay. Should be here with me. Okay.
Reviews from there.
Bismillahirrahmanirrahim. Ladies and gentlemen, from the comstech headquarters, as well as in online in OIC countries. Assalamu alaikum and very good morning. So these students here, the participant here in comstech, so I'm in front of you. However, the participant in online, if you have any questions, comments, or any difficulty, please communicate within chat box so that we can make more active, more interactive, and more fruitful. So I'm here for you. So I'm not the master, but I know a little bit. I will share my experience and expertise with you. And it will be not monopoly. I am the only giving the lectures, you are listening to the lectures. No, it will be in dialogue system. So we'll share our experience, and in between you can ask any question, many questions. It may be science, maybe non-science. It is long duration, 3 hours. So if you feel sleepy, let me know. I have some medicine for I have some medicine. Okay. Yes. Online students, online participants, can you write me, either you can hear me clearly or not? Just if it is, can you hear me clearly? Write yes. If not, write no. Can you? Yes. Okay. Thank you very much. Thank you very much. Thank you very much. Rakibul Hassan, Shundul Javed, thank you very much. There is some voice distortion. Now it's okay. Now you can hear me clearly. Online student, online participant, please. Now it is okay. Oh, it's okay. Everyone. Okay, everything is okay. Take it. Thank you very much. So in this workshop, I'll give 3 lectures. Today, tomorrow, and day after tomorrow. So development of animal models. Development of animal models for metabolic disease. So as discussed, so nowadays, so urbanization, modernization, westernization, Coca-Colaization, McDonaldization, all the things move towards metabolic diseases. So metabolic disease is not only the Western population, Western people or Western everywhere. Now, metabolic disease is common problem and more prevalent problem all over the world. With this emphasis, we focused in the diabetes or metabolic disease is very rapidly increasing in this all over the world, especially the OIC countries. Therefore, we choose these topics. So welcome to my presentation of development of animal models for metabolic disease. So this workshop will explore the role of animal models. Here were some biomedical research, pharmaceutical research, and capabilities in predicting drug efficacy, safety, and toxicity. And most important, last one, it's the training of human development. Okay, how to handle animal, how to work on animal, how to live with animal, how to live with animal. So, training, it's normally, so the animal model training is very unique, not available everywhere. So take this advantage. So before going to my lectures, I'm grateful to comstech. Thank you, Kazima and Maham, and of the comstech team for arranging this beautiful and timely needed workshop. As you know, already I'm from Bangladesh. Okay, this is the map of Bangladesh. Okay, it's a very beautiful country, nicely country. And this is the only one country, few of the country, because we have normally all over all there is mostly 4 seasons. Many countries has 2 seasons, but we have 6 seasons. We have 6 seasons. Every 2 months, we have another seasons. So this is the Dhaka, and my hometown is here. This is the, if you consider this is Dhaka, this is my hometown. We call it Lal Moni Dhak. Lal means red. Okay, Lamoni means the good people living there. So my hometown is a very unique place in the world. In a sense, if you sit here, this is Lal Moni Dhak. So in Bangladesh, this side, and this side. If you see, consider here, India, India, India. 3 sides is surrounded by India, and only this side is Bay of Bengal. The 3 sides surrounded by India. But you sit here, very interesting in my hometown. It is from my hometown to India, it's only 8 kilometers. So I'm periphery in the of the country. So beautiful thing is that okay. You see, in my hometown, there is some places it's belong to India. In Bangladesh, some places it belong to India. And India, some places it belong to Bangladesh. We call some places, we call it Sit Mahal. Okay. So long time ago, before it is India, Bangladesh, Pakistan in the only one country. In 47, okay, it is belong to separate India and Pakistan. So this some territory in Bangladesh, some territory from India, and India, some territory from Bangladesh. But nowadays, the problem is solved, but no more here. But however, in this sense, this is a unique place, my home country. Okay. So during my graduate from hometown, and after that, I studied in biochemistry and molecular biology. This is molecular biology department, University of Dhaka. This is the largest university in Bangladesh. It's the #1 private, #1 public university in Bangladesh. And after my graduation, I worked here. We call it BARDEM, Bangladesh Institute of Research on Diabetes, Endocrine and Metabolic Disorder. So this center, it's all largest diabetes center in a sense. So in a sense, every day, more than 3,500 patients came, diabetic patients. It is the largest center in a sense, all over the world. There is a hospital. There are many hospitals. In this hospital, there is some special wings or small wings for the treatment or management of diabetes. But this hospital is 600-bed hospital. It is focused only on diabetes. But however, other diseases also treated, but it is focused mainly on diabetes. So every day, 3,000, I worked there more than 7 years. Then in Japan, I went to Japan for my nanobachelor scholar and different molecular genetics. I did my PhD from 2000 to 2005. This is the beautiful building here. So in the 10th floor, my building was there. Okay. Then after my PhD, I came in Dhaka, and then on a special invitation with Dr. Iqbal Chaudhry, I came in Dr. Panjani Center and Molecular Medicine and Drug Research, University of Karachi, and I established the molecular diabetes laboratory there, as well as I developed some things for the first time in Pakistan. One is the non-obese diabetics model I developed there. And for the immunohistochemistry, triple channel immunohistochemistry, triple channel immunocytochemistry I established for them. And one of the unique features, advantage of this center is we develop islet isolation techniques for first time in Pakistan, and it is a unique technique, a very unique technique for diabetes research. So tomorrow and day after tomorrow we'll discuss I'll share my experience, what are you doing, what we did there. And then during just before COVID, I was on leave in Dhaka. But unfortunately everything is shut down, lockdown. So I joined Prime Minister University. Then at present, I'm in Dhaka International University, working there. So now come to the point, development of animal models for metabolic disease. So animal model is very important, not only for metabolic disease, for any disease. Okay, so today from this workshop, so we'll focus on what is the basic concept of disease in animal model. how to develop an animal model? What is the advantage of using animal model? Role of animal models in biomedical and drug research. And finally, we'll focus only on disease, metabolic disease. And metabolic disease, there's many diseases, but we'll focus only one, which is diabetes. Okay. And finally, the toxicological investigation on disease model. If you make the disease, if you make any model, either we try to make it the good model or best model, but if it is making the model, it makes some toxic effect, it is no use. Okay, as I told you, so everything has a side effect. Either it is food, medicine, drug, everything has a side effect. If you are making model, but it is make it itself, some drugs are itself toxic. So if any model has toxic effect, it is no use. So animal models, laboratory animals models, and animal models. My question is, okay, human, we are animal or not? We are animal? Yes, no. Are you sure? No, we are human. Okay, so your question, so now, how many humans are there in your country? So some of the students, some of the participants here from Pakistan, okay, some of the other countries. Can you write down how many humans in your country, as well as in the online participant? Can you write down how many humans in your country? So Rashid Hussain, he told me 25th crore in Pakistan. 25th crore means 250 million. Okay, just see it here. Someone write from Pakistan, the 25th crore in Pakistan, 18 crores in Bangladesh. Okay, just we're focusing one country. So in Pakistan, say 25th crore. That means 250 millions. Okay, you are agree or not agree? No, I don't agree. It is not human, it is population. 250 million is the population. Okay, or we call it human population. Only hundreds or thousands and millions are human. So if you are human, so to be a human, what we do? Your parents send you in primary school. After primary school, you go to the secondary school. After secondary school, you go to the college. After college, you are getting university to become a human. Okay, there is a difference between population and human. So that means it takes a long time to be human. All human are population, but not all population are in human. Got it clear now? So it takes long time. It goes through a long process. Okay, same is true if you consider here animals. Okay, one is we call it animal, another is calling animal, laboratory animals. Animals, okay, animals, sometimes animal is in the wild animal, sometimes they're in forest, wild, anywhere, but sometimes in household there is animals. So what is the difference between animals and laboratory animals? So the difference is animals and laboratory animals. So initially, what the scientists or people do, if you I'm giving a practical example. So if normally, so dog or cat are dangerous. Many people like it, many people don't like it. Many people love it, many people don't love it. So especially the babies, they're very much afraid. So if you take a dog at your home or cat at your home, so next, after sometimes after 3 months, 6 months, 9 months, 1 year, okay. So it became loyal to you. Then it become familiar to you. Then the pups, the cats, would very friendly with child. Okay. Then even the child, initially, it is very much afraid of this kind of animals. But after one generation, two generation of the pups, it become part of your family. Your family member, your son or daughter can play with the pups. If the pups grown up, the next generation now it is very fast familiar. You can initially keep far away from the animals. Now, if it is a good cat, you can take it and can sleep with the pups. So after few generations, initially it is animal, it is become a family member. Same is true. The laboratory animals or animals, any vertebrate animals or traditional laboratory animals, agricultural animals, wildlife, aquatic species produced or used in research, testing, and teaching. So, how the laboratory animals develop? Normally, if scientists, people take animals from somewhere, okay, collect it. Maybe some 1 or 2 male or 1 to a female. Then keep in animal in good condition, good environment, good food. In this sense, so they become, they after sometimes they give some pups, their babies. So this is we call it first generation. If the babies came, the first generation, then second generation, third generation, fourth generation. In this way, generation 1, generation 2, generation 3, generation 4, generation 5, 6, 7, 8, 9, 10, up to 20. After this, the pups or the animal is developed. Normally, if you consider today you are here, so you can only recall your father's name, your grandfather's name. So you're very few people knows grandfather's father's name or grandfather's name, or where they came. Okay, that means in human generation, your father, your grandfather, it's 100 years story. You only know the 100 years story. Before 100 years, you don't know where you came, where your father came, where your grandfather came, you don't know. Same is true. The animals, after 10 generation or 20 generation, the things, this is their home. This is the environment. This is their good. Okay. So this big difference of animals, wild animal or animals or laboratory animals is, so after 20 generation, 10 generation, they become very family homely. They're non-poisonous. They're non-hazardous. This is the beauty of animal, the laboratory animal. So laboratory animal and animal, same rat or same mice, same strain. If it is wild, and it is after many generations in the laboratory. So the gene may be the normally human is ecogenetical. Okay, so it's genetics 50% play important role, environment play important role. So genetics is the same, but environment, it makes your life, okay, makes your life different. Okay, environment makes your life different. So the animals long time in the laboratory, good food, good mood, good environment, it changes their lifestyle. It changes their behavior. Okay, so after many generations later, the animals develop non-poisonous, non-hazardous. We call this is the laboratory animal. Now you got the idea what is the difference between animal and laboratory animal. It takes time. So as I told you, to become a human population and human is different. Okay, from 250 million population, human is few millions, few thousands. It takes long time. Primary education, secondary education, college educations, university education. Even you are not humans. Therefore, to become a human, to be a more good human, you are participating in this workshop. Every day you are developing skills, knowledge, everything. So it takes long time. Same is true for the animals. it takes long time to be a laboratory animals. Okay. Yes, online students. So the concept is clear. Yes, sir. So the students participate here. Sometimes we ask the online students, we have to consider this one. Okay. Many students are in the online, so we have to also consider there. Okay. So we have a lot of time here during tea break, lunch break. I'm available here. Okay. If you have any questions, you can ask me anytime, during my presentation as well as after my presentation. Yes, sir. Okay. So the laboratory animals are poisonous. Can you, online student, can you write the question? What is the answer? A or B? A or B? And please, the physical participants, write your answer, either A or B. Which is correct? Laboratory animal. Please write here. Please write. Please write. Please write. Okay. So B. Yes, yes, online students and physical students. Okay, both are wrong. Both are wrong. Both are wrong. Laboratory animals are non-poisonous, non-hazardous. Okay, then we're taking you a small laboratory animal. We call it small laboratory animals. So from the name, small, it is very, very clear. The animal size is small. As I already told you, laboratory animal, they're small in size. If small in size, then easy to handle. Okay, so normally to make the animal in animal house, it's costly because every day you have to keep good food, water, and for their management, you need some people who will take care of this animal. Number one. Number three, the animal lives in a cage, and some good bedding. So if a small animal, what is the advantage of using a small animal? So a small animal in the cages, we can put many animals in a single cages. It takes less space. So in many laboratories, many universities, space is a big problem. So in that sense, laboratory animal or a small laboratory animal, it's a good choice. It takes, if its size is less, it takes less area. It takes less food. Okay, this is the advantage. Easy to handling, short lifespan, and cost-effective experimentation in the laboratories. Those animals are small in size, we call it small laboratory animals, not the beauty. So we already told, okay, animals and laboratory animals. Now, in small laboratory animals, okay, mice and rats, all over the world, mice and in experimental condition, in laboratory experimentation, mice and rats are used for the animal experimentation 95%. 95%. Only 5% all the animals. 95% used in the mice and rats. Sit here. Okay. So animal used in research, mouse and rat. This is the two major things, and they comprise mice and rats. Okay, 90%, 90%. And we also use guinea pig, hamster, and some normally mice, rat, guinea pig, hamster is called rodent animal models. So some non-rodent animal models: rabbit, monkey, cat, dog, and others. We also use monkey, also use donkey, we use zebrafish, we can use butterfly, we can use Drosophila for the biological or biomedical or research. Okay, it depends on your experimental needs. It depends on your objectives. It depends on your aims and objectives and experimental aims. Depending on your aims, depending on the objectives of your experiment, we use which animal is suitable for you. Number one. Number two, one is disease model. If you make a disease model, this kind of rodent or non-rodent, this one. But if you want to the developmental work, this kind of model is not needed. Developmental work mostly done in Drosophila. Okay, Drosophila or C. elegans. This is more better, it's more easier for the genetic analysis, development analysis. However, for disease analysis, animals are mostly used. So here I told you the mouse, rat, guinea pig, hamster, they're called rodent model. Why they're called rodent model? Because they have some kind of narrow edges teeth. Okay, narrow edges teeth at the front of the mouth and adapting the cutting. So they have two teeth there, narrow edges. Normally, therefore, the mice and rat, they can eat everything except chair and table. Okay, so sometimes in your home, in your home or in your office, sometimes valuable documents. Okay, so I don't know if they have good food. If they don't have, they can take anything. Okay, sometimes the animal eat their own baby. Okay, and because this nature, they can sharp teeth, do and bite, and this is therefore they are called rodent. And little by little we see it how we can care and use of small laboratory animal. Okay, appropriate care. One is housing. Okay, housing, good food. Good food is not enough. It is need housing. That means if your bed is the best medicine for you. If you sleep, good sleep is the best medicine, not for any disease, for all the disease. Therefore, sometimes people are suffering from many diseases, complications. You are going to hospital. What happens? Doctor gives some things, medicine or this one, or saline this one with some sleeping medicine. So if you sleep on 2, 3 or 4 hours after sleeping, you feel better. So same is true here. So if animals can sleep well, because we are normally day and night cycle. They have also day and night cycle. So if they have the good sleeping, their physiology will be disturbance. If you don't sleep on 2 or 3 days, okay, sometimes if you don't have a good sleep at night, then what happens next day? All the works, your activity, your mood will be disturbed. So good sleep is a prerequisite to function your routine activity, normal activity smoothly. If it is not good sleep depends on many things. Good sleep depends on many things, but your bed, your pillow is more important thing. So bedding, mattress is more important. Same is true for housing. The bedding is more important for the animal. Management to the animals for scientific research. Okay, it includes ensuring welfare, suitable housing, nutrition, veterinary care, and ethical use. Ethical use means there is some ethical point of view to use animal. So I'll discuss later. Now, come to the point, the character features of commonly used animal models. Commonly used animal model, as I told you, mouse and rat. This is the two of the extensively used animal model, widely used animal models everywhere. Either it is academic as well as in drug development. Let's see some features. Normally we call it mouse. Okay, its scientific name is Mus musculus. Okay, so if you consider mice and rat, they are occupied 95% of research. Out of this, if you consider this is 100%, okay, mice and rat, 100%, only mice occupied 95% research only in mice. What is the advantage? 95%. All laboratory animals, okay, Mice are commonly selected in animal model for variety of reasons, including small size. Small size, it makes facilitating less housing, easy to maintain, short reproductive cycle and lifespan, generally mild tempered. Okay, they're mild tempered. Okay. For the possibility of genetic manipulation. Mice are also used for human disease model, including metabolic disease, diabetes, obesity, Parkinson's, and behavioral study, aging, nutrition, genetic manipulations. So this is the facts and figure of in mice. You see it in mice, we call it one is called inbred mouse. Inbred mouse. Inbred mouse. Anybody knows what is the meaning of inbred mouse? Okay, so the difference between human and difference between animals. So if as I told you, so if we collect one mice on one rat, one male, one female, so their baby, maybe if they give 10 pups or 10 baby, out of 10, there is ratio are 50-50, 5 male, 5 female. They're brothers and sisters. Okay, so if they met, okay, brothers and sister, sometimes brother and sister are all or with their parents. This is the difference between the human biology and their biology reproduction system. So if mating with brothers and sisters or mating with the parents, it goes 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, up to 20 generation. We call it inbred mice. After that, the genetic, all the things are homologous. All the things are homogeneous. For genetic study, this kind of mice is important. This is inbred mouse. Or what is the outbred mice? Outbred means one from here, one from anywhere. Okay, they are not related to, they are not siblings. They are just not siblings. This is the one with inbred mice, another is outbred mice or animals. Okay, this is the difference. So for genetic analysis, inbred mice, it gives a good opportunity to do work or for the research. So it takes 20 more generations, sister and brothers, or parents and offspring. Okay, and outbred is unrelated animals. Okay, so sometimes this is the one is inbred strains, one is outbred stuffs, and sometimes genetically modified mice is knockout or knock-in. Okay, so mice is available. So this is in inbred mouse. Inbred mouse for genetic analysis is okay, good that identification of animals. But in outbred mouse, normally, especially the non-obese mouse, non-obese diabetic, this is the example of outbred mice. So let's say some features. Normally, if adult, their weight is normally 20 to 30 gram, 20 to 30 gram. Okay, the lifespan 1 to 3 years. Here, you see it here. Their heart rate and respiration rate, small animal, the heart rate is 300 to 800. What is the heart rate of human? Anyone? 72. Yes, online student. What is the heart rate of human? 72. And heart rate of 72, and there is 300. So this small animal, the heart rate is almost 5 to 6 fold, 7 fold. So therefore, is their metabolic rate is higher. They're more active. They're more active. Therefore, so to catch a rat or to catch a mice in open space, at least 5 people is needed. You can't. Okay. And water consumption 3 to 5 ml, food consumption 3 to 5 gram per day. So mice breed continuously throughout the year unless conditions are very unfavorable of them. And sometimes light and day cycle. In the animal house, normally we make it 12 for light and 12 for dark. Otherwise, any light during night time, if light is there, you sleep is disturbance. Therefore, in the animal house, we make the light. In this, there is some programming. This one after 8 p.m. Okay, 8 p.m. or 8 to 8, or sometimes it depends on laboratory. Okay, 12 hours should be on dark. Even if it is dark, mice can be bred using normally the what is the breeding system? Normally either one male, one female, option one, or we call it one male, one female, okay? Monogamous or polygamous. One male, more than one female. Normally, what we in practice, what we do, we make one male, two female, or a practical application. So mouse pups, they're hairless, blind. Okay. Sit here. These reproductive preferences. Okay, in mouse. Normally it takes 4 to 7 years. Near gestational time is 19 to 22 days. It makes easier. Okay, it makes easier. So shorten gestational time, it makes within shortened time. Normally, if you want to work on large amount of animals, then the litter size is 9 to 12. Okay, normally human after 1 year, you have one baby. But in case of animal, in case of mice, after 3 months, so you get at least 9 to 10. So normally human is one baby after 10 months. It is common. Sometimes 2, sometimes 4. So in animal, so if you got at least 4, sometimes many animals gives 9 to 10 is normal. But if you can animal gives 4 pups, you can use it for experimental purposes. But if any model, any mice gives less than 4, so these mice may have some problem. Okay, so if less than 4 animals, those less than 4 pups, it is better not to use this animal for your experimental purposes. So the mice are nocturnal animals. It means they are active at night. They're hyperactive at they're active at night, hyperactive at midnight. So sometimes if you see an animal house, you will see all mice are sleeping. They can normally, because especially they are. So as I told you, so it's very difficult. Okay, it's normally so many things during the environment, many things change. But normally we have a biological clock. We have a biological clock. Normally in the daytime we work, at nighttime you are sleeping. Either in the morning you wake up, some people use alarm clock, and when is alarm is alarming, running, make it off. Or many people, most of the people of the world don't use alarm. Then how to wake up in the morning? Our biological system or biological works. Okay, so normally, say they're active at night and more active at night. And they're very sensitive hearing. Mice are very sensitive hearing. And therefore, they can eat everything. But they have sensing, hearing is so important, they can differentiate. Either it is poison or not poison. This is the big difference between human and mice. But smell, you see, in the airport some 10 dog, 10 animals, they can by seeing smell, they can understand if anything is there or not there. Human can't. So this is the difference. One is animal and one is human. Mice, they have very sensitive. And aggregation, normally, mice mild to moderate. Okay, it is normally they are not aggressive. In extreme cases, in very extreme cases, they are very aggressive. Okay, sometimes it depends on many things. If you are in family members, if you have 4 brothers and you have many brothers and sisters, sometimes what happens? Okay, sometimes what happens, your brothers and sisters some fighting. Same is true. Okay, if all are calm and quiet, possibility 1, if you have more brothers and sisters, conflict of interest, only good food goes to the brothers, Okay, then brothers and sisters, there is conflict. Okay, and many things, the same is true if they normally if they live in cages, few animals, sometimes they exist. So, online and offline, please write your answer. Mice blood volume is 1 ml, right? 5 ml, 10 ml, 20 ml. Yes, online student, please. C, 10 ml. Dr. Tariq, 5 ml. Umar Rafiq, C, 10 ml. Okay. C, 10 ml. 5 ml. Okay. C, D, 5 ml. B. Is anyone from this one? Someone like A. How many agrees on A? Please raise your hand. No one A. How many agrees B? Okay. How many agrees with C? Most of the people agrees with C. How many agrees D? No one. Now, I just want to, yes, online students, check it. Just please wait. Please wait. Can you, everybody, all of the students here or online, you know what is your body weight? Almost all of you know your body weight, right? Please write your body weight in kilogram. Okay, Johan Sharfraz Ali, Muhammad Usmani, they wrote A, someone wrote A and C. Okay, all students write your body weight, right? And what is the total volume of your blood?
5 to 7% of the body blood.
Yes, blood.
5 to 7% of your body weight.
5 to 7% of your body weight. Please calculate. If you have 5 to 7% body weight, what is the total volume of your? Okay, my question was, what is the total volume of blood in your body? You know, total volume of blood in our body? How many liters? 10 liters, 50 liters? 5 liters. Okay. Okay. If you have 5 liter blood and your body weight is 60 kilogram, can you calculate what is the percentage of your body blood? Yes, madam. If it is equal 5% of your body weight. Sorry? 6 liters. If it is equal 5% of body, so you are not correct, madam. You are not correct. Not 5%. If you have 5 liter blood and your weight is 60 kilogram, okay, then what is the percentage?
8 liters means for the 60 kg, if we calculate 5%, it comes around 8.3 liters.
Make it very simple. 5 liter blood, the person weight is 80 kilogram. If someone has 80 kilogram and 5 liter blood, what is the total volume? Okay. Just remember this one, 8%. Okay. So the mass blood volume is number 1. It is A, 1 ml. It is weight is normally mice in adult mice weight is 25 gram. In 25 gram, okay. No 25 gram. If this is the normally the whole. If it is 20 ml, it's impossible. Okay. 10 ml just half. If it is impossible. Only possibility is 1 ml and 5 ml. So normally, okay, it's almost 1 ml. Okay, 1 ml. We can collect blood. Okay, one of like very similar to 1 ml. So now come to the point rat. Okay, come to the point rat. So rat had humans a long history of coexistence. Rat constitutes one of the most commonly used laboratory species, second to mouse, after mouse. Yeah. Okay, and you see, normally, as I told you, the laboratory animal develop in the laboratory. So in the rat, sometimes we call it oyster rat. Okay, so many people make a confusion here. So sometimes in the nomenclature is belong to the scientist name, but the oyster laboratory, there is laboratory in USA. So how the animal model is, how the laboratory animal develop, they took animal from somewhere, embedded in their lab, 1, 2, 3, 4, after many generations, this developed the rat, and this is the most extensively used rat all over the world. Oyster rat. It was developed in the laboratory, oyster laboratory. What the beauty of this one, like mouse, that possesses a number of qualities which makes easy to high suitable animal model, including small size, small in size, known genetic background, short generation time, similar to human disease. Okay, and sometimes we call it known microbial status. And rat have been used for animal models and numerous diseases in case of nutrition, genetics, immunology, neurobiology, infectious disease, metabolic disease, and behavior. The largest use of drug discovery, efficacy, and toxicity studies is done in rat. Okay, so as you know, so for any drug discovery, it goes before going to the clinical trial, it goes for the preclinical study for clinical safety, efficacy, and toxicity. So in toxicity testing, it should be one small animal and one large animal. Okay, so last point you see it, and rat rarely binds to their handlers. Okay, mice and rat. In our experience, I worked a long time, so I never ever faced any problem. But I found only one student of my on our institute. Sometimes only one day the mice bite. Normally rat never. So working with mice and rat, so if you're kind enough, treat the animal in a humanized way. Okay, humanized way. So if you understand their behavior, don't use them, don't abuse them. So it is animal. So if you're just going to animal house, hold the mouse or hold the rat and give you medication. No, there is no shortcut. Okay, so normally this animal should be treated as your son or daughter. More, if you get more love and affection, then it will be friendly with you. You can do your test very easily. Otherwise, if it is aggressive, either mice or rat, so this, if it is aggressive, either the effect of your drug or effect of your medicine, it is due to your drug or due to his behavior, it's difficult. So make them rat, don't make the animals aggressive. So normally for any research, normally if you make any disease model, so many genes are part of the gene have very homology with mice and rat. Therefore, for genetic study, we can use both of them. So rat genome database is there. So if you see what is the, if you find any of your gene, either it has relation to the rat or mice, then you can predict something. It is 50% similarity, we can work on it. At least 50% similarity should be there. So the basic facts and figures, male, and this is the weight is normally in one rat. Normally we call it near 200 gram. 200 gram. Sit here, its respiratory heart rate is also, it's higher than human. Yes. Okay. Lifespan is 1 to 3 years. Okay. And same is true here, male and female, and gestational time is 21 to 23 days. It's very similar to the mice. The way we mating the mice, same is here, either one mice, one rat, one male, and two female in this way, this one. So the rat pups, rat pups is the rat pups figure normally initially. So this is the difference between the developmental mice, rat, and human, and so cow. So human development happens before and after birth. Okay, the cow development, all the development was done before birth. But here, initially the pups, they're blind. So development is not completed. Okay, so normally, if one cases, 1 kgs. So we found 1 kgs. This is our own experience. It's many puffs. Okay, initially they are lactating. How? They couldn't understand their mother, this one, but only the smelling power. The smelling power make them so sensitive. They are hairless. Initially, they are pink color. Okay? Pink color. Anybody has idea what is the weight of the pups, of rat pups? Weight? One to 3 gram. Answer is no. Any other? 5 gram. Very close. 7 gram. 6 to 7 gram. 6 to 7 gram. So I'm showing you the pups because so sometimes some model development you have to give the treatment in pups. So this is the beauty. This is the beauty. Some model. It is not in adult rat. You have to give, if you the adult rat, same chemical. If you give adult rat, it may produce different disease with different pathophysiology. If you give the same drug to the pups, it will make the same disease with different pathology. Same drug, 2 different rat. One is pups, another is adult. Will make different pathophysiology. So tomorrow, inshallah, we'll discuss tomorrow and day two how it happens and how it happens, what is the mechanism and so on. So what is the difference between mouse and rat? Size matter. One is mice, its weight is adult one, mice is 20 gram, make it very simple. One is 20 gram, another is 200 gram. That means 10-fold higher body weight. If so, normally in the mice 2 to 4 inches and rat normally 9 inches long. Size matter. So in case of mice and rat, in same cases, okay, more mice can be accumulated compared to the rat. And mice and rat, another big difference is tail difference. Tail is very important for biological research. So in mice, the tail is thin. The mouse tail is thin. But the rat tail is heavy tail and thick tail. It makes a good opportunity to do the biological research, especially the metabolic disease. If you consider here, okay, a long tail, okay, and short sharp tail, okay. This one is the rat. So sometimes we want to check that if you give something, or especially either it is checking the your model diabetes, we have to check blood glucose in 0 minute and 120 minute. Sometimes we do 0 minute, 15 minutes, 30 minutes, 45 minutes, 60 minutes, 90 minutes, and 120 minutes. So if you want to check or collect blood in different time point, okay, so in that cases, rat is suitable because you can collect from blood hair, you can check from, you can collect from blood hair, you can collect from bear hair, different time point. So my student had to do 4 to 5 times from the tail vein, they can collect different point. So this is the beauty of rat. From the tail vein, you can collect very small amount of blood and you can check some parameters. Tail, it called capillary blood. So the rat blood volume is, yes, online student and physical student. Who's on correct? 1 ml, 10 ml, 50 ml, 100 ml. All are right. Yes, online student. Rat blood volume is 10 ml. Okay. No, someone write 1 ml. 10 ml. 10 ml. Okay. So why I'm asking you the blood volume of 1 ml and 10 ml, it has a meaning because during the drug, if you give the drug somewhere, normally in case of, we do experiment in vivo. So when you consider the in vitro, normally in vitro test, normally we do in test tube. In test tube, normally we calculate concentration, nanomolar, micromolar, millimolar. So when we got, so we are using some nanomolar, micromolar, millimolar. And after that, we get good result, and we, after doing many tests, we giving good result with some doses. Then dose, normally this we convert these doses, in vitro doses, to in vivo. For that, in case of mice, the drug will goes 5 ml blood. And in case of rat, your drug will goes to the 10 ml blood. So this calculation, this amount, this 10 ml of 5 ml is very important, 1 ml of 10 ml. So during my lectures on in vivo correlation, I tell them what is important of 1 ml. This must be take consideration. The rabbit, okay. Yes.
Mice to the rabbit. So you have told us that most of the time for the rat animals, the mother, we use mice. So why it is
Sizes matter. So it is size, because it less size, easy to handle. As I told you, in mice, weight is 20 grams. Rat is 200 grams. That means, as I told you, if you normally once you have a mice, that means at least 3 years normally. So day 1 to 3 years, you have to keep these mice at your laboratory. So mice take 3 to 4 grams food, and rat takes more food. So normally the animal, we have to purchase some food for the animal. You have to, every day you have to give the food. So it cost effectiveness. If it's normally the food is take 4 mice or 5 mice food, 1 rat. So it's the cost effectiveness. Okay, you can do it anywhere, but it's cost effective. Ultimately, anything goes. If some of the mice take less food in 1 day, 30 days, 1 year, either you use it for any experiment or not, it used in an experiment. The mouse should be alive. It should be kept in your laboratory. Okay. Economical. Yes, madam.
So mice and what? Mice. Is this right?
So in
That's why mice.
no, and no, it is not always correct. Not always correct. It's normally. Wait, okay, wait. Yes, please wait. I have a slide. You can get the answer. Please wait. If not, it depends. It depends on gene to gene. It depends on the. Please wait. This is the rabbit. History of rabbit. Okay. So they have been interesting and very important role in biological research. The Louis Pasteur used in the rabbit for the vaccine development. There is a rabbit we call New Zealand rabbit. Okay. Normally, the model hyperlipidemia is hyperlipidemia, dyslipidemia, and atherosclerosis. All the things are normally this kind of model is developed in rabbit. So now my question is, what is the difference between hyperlipidemic and dyslipidemic? Anyone can tell me hyperlipidemic and dyslipidemic?
Dyslipidemic is a general term that may be hyperlipidemia.
And what is the hyperlipidemic?
Hyperlipidemic increased concentration of lipids in the body. That is when you have to push the hyperlipidemia.
Okay, rabbit have been used as a model of human pregnancy. Sometimes, normally, most used in rabbit for the polyclonal antibody development. Normally, what we do, if you want to work on some new genes, gene is there. If you consider this is the unique genes, so if gene is there, now expression is the protein. So if you work in the first time in the research, so no proteins, almost many protein has commercially available antibody. However, those are novel proteins, so no one is working on it. So this antibody is not available. So what we do in that senses, we normally express this protein in some E. coli system or yeast system or mammalian system. We purify the protein, and with this protein, a purified protein, we injected to the rabbit. So after sometimes we give second dose. So normally during the COVID, almost all of you give vaccinated dose 1. After 1 or 2 months later, you give good dose 2. Then after some time, you give another booster dose. Same is true for the antibody development. The protein, purified protein was give the rabbit dose 1, then dose 2, or normally we give 2 doses, sometimes booster doses. Then what happens? You give the vaccine for the COVID. So in your blood, you will find COVID antibody. Same is true if you give any antigen to the rabbit. So after 2 or 3 months later, in the blood, we'll get the antibody of this protein. We call it antisera. So mostly antisera is developed, or polygonal antibody is developed in the rabbit. So what is the beauty? Normally, as I told you, mice has 1 ml blood. Rat has 10 ml blood, and rabbit has 60 to 200 blood. So my question is, so if in human blood, sometimes if you collect the blood in clinical test, if you centrifuge 10 ml blood, what is the volume it will expect in plasma? 10 ml from human blood, 10 ml blood, what is the volume of plasma? What is the from 10 ml blood? 10 ml blood, normally for safe side we are making 50, so it's 5 ml. Okay, 5 ml plasma. So human blood, what is the difference between human blood and rat blood or mice blood? Human blood is thin. Mice and rat blood is thicker. Okay, one difference. So in case of human, so 50% or 60% will get plasma. But in case of animal, you will get less. Okay. So this makes the beauty. If you make the polygonal normally, if it has antibodies, so rabbit blood has say roughly 100 ml. So antisera, you get 40 ml antisera. Okay. So students, online or offline, both are here. So I have my practical experience how to develop the polyclonal antibody in rabbit. So if you are anyone interested, there is an antibody that time, it is not commercially available. So we make an antibody KA. We have antibody, its name is KARH4. There's KA Khalek Abdul and this RM Rahman Hafizur 4. We make this antibody. So rabbits are routinely used for atherosclerosis. This. Okay. This is the some characteristic feature of rabbit reproductive system, male and female. This is the weeks, litter size. Normally, mice and rat, they give 10 or more than 10, and 10. Normally, they give baby 4 to, okay, 4, 5, 6, 7, 8. Okay. Then another is a guinea pig. Guinea pig. This is mostly used. Okay. So used for antibody production, many disease model. Okay. So a small guinea pig is small, it's stocky. Okay. So this guinea pig, okay, play important role. Many Nobel Prize awardee studies. Okay, the animal normally small animal to small to normally rat weight, mice weight is 20 gram, then 200 gram. It's little bit increasing body. Guinea pigs have also been useful tools for cholesterol metabolism, asthma, placental development, Alzheimer's disease. Guinea pigs have many similarities between human, hormonally, immunologically, and physiologically. The experiment was done, the vitamin C experiment was done. Normally the disease, it was done on guinea pig. So now, up here, we discuss about the animals and laboratory animals. Now, we want to make some, what is the model? What is the model and what is the animal model? Okay, see, this is the photographs of human. So, the human in Asia, the human in Africa, the human in America or Europe, physiology is different. Physiology is same, but digestive system is same, but appearance is different. Okay, appearance is different. In sometimes in same country, in northern part to the southern part. There, everything is different. Language different. Food is different. Okay, it may sometimes in same singular country or in the same country. So even by seeing the structure, you can understand either if north or south. Okay, here many people by doing anything, you can some gaze. Maybe he's from Asia, maybe he's from Europe, or he's from Africa. By seeing the face. But what is the difficulty here? Is it here? This is human. This is the human, and this is the mice. Okay, mice or rat. Okay. So what is the difference? Human are different. Mice are the, because if it is inbred mice, it comes from the inbred system, homologous system. But human is different. Human is different. So this is another problem. If you want to make some disease model, it may or may not mimic the human system. So problem is everywhere. So during tea break, one of my participants, my madam told me they're facing some problems. Okay. So facing some problems. It's good because when he came here, so the Muslim brother sister, I told you little story. Our prophet, you know, he was did long times when he became a prophet, because he was in Guha, some places, very safety there, security there, no tension for the food, this one. After that, Allah Ta'ala ordered him to go outside. Okay, to go outside. Then he's crying, so sir, I'm fine here, I'm in comfort zone. No, you have to move. So in Prophet in small places, if he's there, he'll be prophet. He'll be not the best prophet. Then Muslim people will be not all over the world. Only Saudi Arabia. Then what happens? He came in at home. He invite peoples. He faces many problems. Then the problem is too much. He left his hometown. From hometown to Medina. Then again he faces many problems, but however he overcome the problem. After that, the Islam is all over the world. Same is true, madam. So if problem is there, then we have grow a thinking ability. You have think in many ways. So if in comfort zone, as I told you, in population and human, when at your hometown with your father and your mother, no need to think anything. Food is ready, everything is ready. But once you are in out of hometown, everything is difficult. Everything is unknown. Environment is unknown, city is unknown, language is unknown, food is unknown. But if there is, there is a lot of problems. But you will stay at your home. All the successful men, all the Nobel laureate, all the successful men behind the civilization, all left their home. They know there is sometimes outside the country or outside the city, there is a problem. If you go outside the home, you think how to overcome the problem. So, though there is some difference between human and animal, okay, to making disease or making disease model, it's lots of problems, lots of heartache, lots of difficulties, but there is way. So, as I told you, this is the human and this is the mice experimental models. Inbred mice or inbred rat. Say, if you consider our evolution, okay, if you consider evolution, normally, if you believe on evolution, then this man, mouse, chicken, zebrafish, this man, mouse is very close on civilization. Revolution. It makes sense. It makes a sense. That means there is a many similarities between though their shape is different, Size is different, but many similarities is there. This makes our advantage to make some model, to make some disease model, to understand the pathophysiology of human. At least you get some idea by doing experiment on anyway, because many similarities are this similarities there, similarities also there. If you see it here, in human, we have 46 chromosomes. Chimpanzee, 48 chromosomes. Dog, 78 chromosomes. Okay. See it here, it is human. Okay, in human, if you consider the DNA sequence similarity, DNA sequence, not gene, DNA. Normally, gene sequence in chimpanzee 98 to 80% similar. With mouse, 92% similar. It is the DNA sequence similarity. It makes some logic, it makes some sense. We can work on mice. We can work on mice, or we can work on that. The genetic similarity. So my question is online and offline. Please write your answer. So the DNA sequence similarity between human to chimpanzee is 98%. In mice, 92%. What is the genetic sequence similarity, DNA sequence similarity between you and me? Please write your answer. Yes, online students, if you write your answer, I can understand you are listening my lectures clearly and you can understand it. What is the DNA sequence similarity between you and me? Right, right. Okay, 99.99. You should have that. Yes, Kishore, thank you for joining. 99.99%. And Dr. Farnaz Mumtaz, 98%. No, madam, Farnaz Mumtaz, you are wrong. It is 99.99%. So your similar, your genetic, your DNA sequence and my DNA sequence is 99.99% similarity. Normally, sometimes we call it a small changes make a big changes. Okay, though we have trillions of base pair. If it is 0.001%, it makes millions of changes. Small changes make a big change. Okay, thank you very much. If you see the sequence homology, okay, this histone one, this is the mice, human, mice, rat, cow, this one. You see, most of the things only here one amino acid, here one amino acid, here amino acid. This is the only change. Most of the things are very similar. It makes another logical sense that yes, we can work on it. The best example is insulin. So this is the insulin. This is the insulin. Nowadays, for diabetes, we are giving insulin as injection. It is the recombinant insulin, human insulin. So this study of recombinant insulin started in 1980s. So before 1980s, all insulin it is either from or cow. So the studies pork and cow, it makes very difficult. Sometimes Muslim people is very much, sometimes we have some if it is pork insulin, okay, and sometimes cow insulin. Sometimes from religious point of view, many people don't want to use it. It makes a, though all are insulin are making for the betterment of the disease, but sometimes some religious barriers is there. Many people want to take it, not want to take it, many people don't want to take it. So recombinant DNA technology make the problem solved. But however, so even you are giving till today, so the insulin from animal is available in the market. My question is, so you need human insulin, but before discovering human insulin in market, you are giving insulin from cow or insulin from cow. If you work in how it works, because sequence similarity is it human insulin, pork insulin, and beef insulin. Okay, only in human insulin and beef insulin here only one amino acid change, one or two amino acid changes. It makes the change. It makes the functional changes. It's almost similar. Functional changes, the function of human insulin and function of beef insulin and function of pork insulin almost similar due to the sequence similarity. of protein level. So during my PhD study, we did one gene, we call it DJA4 protein. Okay, this is the chapter. This, you know, the HSP, heat shock protein. Sometimes we call it stress protein. Another is called heat shock protein CO2. This is another class of protein. And this protein we call chaperone protein. This is also chaperone protein. And it's called the cooperative protein. We call it associative protein. We call it co-chaperone. Chaperone activities. So this is me. This is my PhD work. So my work is, so the DJ4. This is the protein. So this is the protein. So this is the gene newly identified, newly discovered. So my work is there. Clone this gene, express the protein, purify the protein. Though it is a partner protein of chaperone, it must have some co-chaperone activities. Please prove it functions as a co-chaperone, then you'll get the PhD. What exactly I did. Okay. So before going there, because okay, see here, the question someone asked me, this protein, heat shock protein 70 and 72. Okay, see here. This is the human to mouse is 97 point, similarity is 97.99.7. Human to rat 99.8. 1% difference. That means you told the similarity is more in mice. It is not always true. Okay, it is 99% and human HSP 70 with mice and 95%, in rat 96%. And HSP 72 in mouse 98.9, in rat 98.6. Okay, I'm giving one example. That means I have 3 proteins. One is HCC 70, that means heat shock protein 70, and the heat shock C means heat shock cognate protein 70. All 3 is molecularity the same. Heat shock cognate protein normally always expressed. Heat shock protein 70 expressed only in hypertemperature, in a stress condition. And heat shock protein 72, it is expressed only in testes. So their molecularity is the same. They have little bit changes, but their genes you can normally, the sequence of amino acid sequence. If it is amino acid sequence similarity, in case of DNA sequence similarity is more. So your question, the answer of this question is not all genes, the similarity sequence is higher in mice compared to the rat. It depends gene to gene. Okay, got madam, your answer? It's not always true. That means maximum similarity will be the mice. No, answer is no. Answer. Okay. Now come to the point of animal models. Now come to the points of animal models. So what is animal models? If you normally, my question is, anyone tell me what is model? What is model? What is model? Can you tell me what is model? What is model? Okay, anyone? So who is your model or who is your role model? Cockpit. Anyone? Okay, she told her model is our cockpit. Anyone? Yes. Everybody with her? So model is a different. Okay, that means so someone has model, he want to be a puppet. Someone who is follow, want to be like his father. Okay? Someone want to be his mother. Someone want to be like Professor Iqbal Chaudhary. In case Dr. Iqbal is his father. Someone want to be sometimes some teachers influence his life, his or her life, in such that we want to be like these teachers. Okay. Sometimes you want to be like Wasim Akram, cricket player. Sometimes many students or many female students want to be a Kareena Kapoor, the Bollywood actress. The zero figure. Okay. If you want to be if your model is Kareena Kapoor, don't take it otherwise. Okay. So it's 0 figures. So if you want to be a Kareena Kapoor, you can't. Because many things you can do, many things you can't do. Because her height and your height is constant. You cannot change your height to her height. Okay. So his hair is this one. Only you are focusing only his body shape. If you have a zero figure, that means model. If you want to do or you want to be this or that model, you can't exactly. Many things you can't, but something you can. Something you can. Same is true in animal and model. Sometimes it's impossible to reflect or mimic all the condition of legend or model to you, but sometimes something. Same is true animal models. So if we impose or mimic some things in the animal, we call it animal model. Okay, an animal model is a living organism in which normative biology or behavior can be studied, or in which spontaneous or induced physiological process can be investigated, and in which the phenomenon of one or more respectively resemble the same phenomenon in humans or other species in animals. As I told you, even though you want to follow someone, okay, because madam, if you told me that you want to be your model is Prophet Muhammad, you can normally if you follow his lifestyle, okay, his lifestyle, so many things it's do and don't. It's almost impossible to follow in modern era. At least you're trying to follow as much as possible. You're trying to follow as much as possible. Okay, same is true in the animal model, in the disease model, because you don't know anything of Prophet Muhammad. You didn't see him. It's part of his nature. It's just advice, good advice. Do. You want to only following this one. Okay, many things is written, many things not written. So if something is doable, you can do it. Something is very difficult, it's impossible for you. But how if you want to follow him, okay, then we're trying to manage, we're trying to follow maximum most of his advice, do and don't. Do this to get good human. Okay? So same is true in animal models. Normally it's impossible to make all the characteristic feature or disease in an animal. So what at least we can do something. Okay, if we can do something. So these things in animal, it's many ways. Either it's a spontaneous system or in induced system. Okay, a spontaneous system is, so you're at your home, you're studying every day. So curiosity of knowledge, curiosity of gaining knowledge, curiosity of skill development, you can do by yourself. Induced means, so you are doing some courses, you're going to school and college, there is an exam. Okay? So if exam is there, that means it is induced. You have to read to pass the exam. Okay, some people pass the exam and some people don't go to university or college, so much, but more knowledgeable than you. They acquire knowledge by himself or herself. Spontaneous. Okay, one is induced. Factors there. So there is two different. One is, so in case of a spontaneous model, how it developed. So if you are here, so one is, the guinea pig, another is mini pig. What is the difference guinea pig and mini pig? Size. So if we put good guinea pig, good food, good mood, good environment, it's guinea pig. But if we make it the restriction of the food, no. So maybe restriction of food, little by little its first generation, okay, its size will be decreased. Second generation size will be decreased. Ultimately, with the size just half. It will be. So same is true in human. If you are here, good food, good mood, everybody is taking Western food, junk food, only eating, sitting and eating, sitting and eating, sitting and eating, little by little will be obese. Okay, if someone restrict the food, then if you have twin baby, One is good food, good mood, eating and sleeping, and someone is active, dynamic, he's taking his food according to his need. Twin baby, one is obese, one is non-obese. Okay, who obeys? See, if she got married, he or she got married, next generation, next generation, next generation, next generation, all will be obese. Chance is there. Same is true how the spontaneous model is developed. Okay, so the spontaneous model is developed mostly normally in case of diabetes or dyslipidemia. Many model is developed in Japan. How they develop it? Say, if you have in the laboratory animal, you have 100 rats. You are checking blood glucose. So normally in this room, we have 50 people. The blood glucose range is 80 to 100 milligram per dl, right? Someone has 80, someone has 85, someone has 90, someone has 95. We'll get a different range blood glucose from here to there. So what we do? So if you got normally the animal blood glucose, someone has 50, someone has 70, 75, 80, 85, 90, 95, 100. So if we take one rat with 100 milligram higher range, one male in higher range, one male in female in higher range. If we mating them, the first generation. So chances there all the pups will be blood glucose will be the little higher. It is not 70. So this higher range of blood glucose male, higher it is blood glucose female, mating needs their offspring chances blood glucose more. This is called generation 1. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. So normally, so after one generation, again they check the blood glucose. So if it is 100, maybe first generation will be 100, 105. Second generation, 107. Third generation, not all animals, some animals. Okay, as I told you, it is normally in pups, 10 pups, out of 10, maybe 1 or 2. Normally 10% positive chance is there. So it is initially 100, second generation 105, third generation 110, then 110, 110, 115, 120, 20, 20, 22, 23, 23, 23, 23, then 25, 126, 130. After 20 generation, the blood glucose of a rat is, the level is equivalent to the dose of diabetes. It takes long time, lots of energy. This is called a spontaneous model. Long time with some environmental change, lifestyle change, it makes a spontaneous model. It takes long time, 10 to 20 generations. Another is induced model. If you do something within very short time, we'll get something. We'll study later what we did. We'll study later. Choosing the correct animal model. This is the most important feature of this. Okay, correct animal model. Either any disease, okay, either disease. So either you're taking rat or mice to make the model, then you have to make a correct animal model. So the model, or you are making some disease to the animal, should resemble or should mimic the pathophysiology of human of the disease or the clinical presentation of human. This is the most important thing, okay, so that you can choose the correct animal model. As I told, good science and good models, good animal care, one is model. To make the model, good model, you need a good care of the animal. Good care of the animal. So, good care of the animal. So, if you make diabetes is a disease of high blood glucose. It makes polyuria, lots of urine. Then you have to take special care, good care for this model. So, the experimental use, one is toxicology studies and bioassay. Normally, this model I used, normally mostly first you have to do toxicology. Bioassay, different normally if we got any compound, any fractions, any extracts, what we have to do, we do bioassay. There is many bioassay, either it is active or inactive. Different condition on, if it is the diabetes model, then insulin secretion activity or insulin level on the bioassay, screening chemotherapy agent, studies related genetic and cancer, and drug action on central nervous system. So animal model can support and help develop medical product development during the non-clinical phases. So this animal model is important because the drug, any drug in the market today, all drugs gone through the animal study. Without animal study, there is no drug in the market. It's prerequisite. Well-designed animal models can address the regulatory safety concern. Because any drugs come to the market, it should do some regulatory affairs. Okay. So animal studies can provide insight and value in the preparation of the regulatory submission. Because in the regulatory submission, there is mandatory. At least one small laboratory animal and one large animal should be done. So these animal models in biological and functional studies, okay, provide understanding and fundamental biological mechanisms, okay, and provide understanding the complex biological problems, serve as a tool of discover and quality impact of investigation treatment, whereas the disease and chemical toxicities, and use for disease research. So experimentally created models either through surgical, genetic, or chemical modifications. Genetically varied models which mimics the human conditions. And sometimes we make it transgenic mice. So by doing some transgenic genetic modification, we can do some transgenic animal. So this is the importance of animal models in pharmacology. So many Nobel Prize awarding experiment or the discoveries. So all in animal models, people use many. You see it here, 186 award researches, the animal models and their project in their 21 are subjected to a scientific project. So mammalian models were it's normally 145 Nobel Prize in Physiology, 144 Nobel Prize in Physiology. Animal model, mammalian models were used. So that means it has a great importance in science, in novel science, okay, science and discovery. How necessary an animal model in modern drug discovery? It is absolutely necessary. Without this, you can't do modern drug. So now see it here, the significance in human health and advancements. Small laboratory animals serve as crucial models for developing human disease and developing potential treatment. So to understand the human disease and the potential treatment, the model is important. Suitable model is important. They enable researchers to gain insight the disease mechanism, which is the efficacy and safety of new drugs. Okay. And the medical advancement and improving the human health. So to either you compound a drug, if you want to do something, either it is safe, test on animal. Safety first. Either it is effective or not. Safety, efficacy, it should be tested on animal. Sometimes we make genetic manipulation. Okay? Genetic manipulation. What we do by doing so from outside, you can give some gene. In this way, you can develop some disease. Genetic modification embedding in necessary study to impact this specific genes on various physiological and pathological process. These animals help us to understand the complex genetic disorders and for the genetic therapies. Nowadays, okay, so there is some kind of advancement technique. So they are giving gene, delivered the genetic system, genetic material to the animals. This gene is tagged with some color. Okay. Tagged with some color, very beautiful experiment. Okay. If it is tagged with green color, all animal will be green. In this way, if it's many animal is there, the beauty is so in diabetes model, so we are giving either it is spontaneous or not spontaneous. Okay, you have to check blood glucose. Okay, either it is diabetes or not diabetes. Same is true for other disease. You have to check some parameter. The beauty of genetic modification with some tag with color. If genetic modification is done, or you are giving some genetic materials, using some systems, if it is done, successfully done, if your animal, it is green color, if animal is green color, then you can easily understand, yes, my genetic manipulation is done. No need to check. This is some kind of advantage. Because the ethics of animal, some ethical issues there. So minimum number and minimum disturbance to the animal, it goes here. Otherwise, you have to check every day or every week. It makes it different. Another is training. This kind of animal handling or animal management, okay, disease model, animal model, it gives education and training to the scientists, healthcare professionals, and providing an opportunity to hand-on learning experiences and help students to develop critical thinking, critical understanding, experimental design, how to do experimental design. Okay, this is the higher education system. So if you think, if you're a student today, because this experimental model or animal model, it takes time. So you can design. If you want to develop some spontaneous model, it takes long time. If you can induce model, so if you induce something today, you are getting that today, 7 days, maybe the acclimatization or familiar with this environment, 7 to 10 days or 1 month. After that, you are giving injection. So either some chemicals, okay, that make your disease. So after 10 days, 15 days, 1 month, 2 months, 3 months, 4 months, or sometimes points, this disease is developed. Then you want to take your, you already developed some traditional medicine. This is available, or some of the part of your country. So in some disease, people are using this one. By what is that people are using for the management of this specific disease. So why he or she is using this disease, he or she don't know. Because his mother told, okay, if this happens, please take these plant materials. So he or she got this knowledge from his father and mother. He or she got the matter from your father and mother. Because that means for the management of disease, it already incurred long clinical trial. Humans use it, it is working. But how it works, don't know. Now, if you want to know how it works in something with this kind of disease, then you have to develop a model and do your experiment on this model. Okay, the students normally sometimes the MPhil student or PhD student, if you can understand what is the composition of these materials, what kind of materials is there, and how it works in this model. To understand the pathophysiology of disease, to understand how your compound or plant materials work in this model. If you do, you can get some MPhil degree, PhD degree. This will come education and training. And you see, sometimes we are doing some experiment in, we call it laboratory work, we call it bends. Ultimately, if you do some work on bends, your product should have some value. Okay, ultimately, we are doing some research. So sometimes we are getting some degree, or sometimes we are focused on to discover some drug or something so that it can useful for the either it is health or education or agriculture somewhere. Okay, we call it bends to bed. So you are working today in the laboratory. Ultimate goal today or tomorrow or day after tomorrow, after 15 years later or after 20 years later, it may go to for the human, for the betterment of the human, or the digital, we call it bed to decide, bends to bed. And if it goes to the veins to bed, you are working today at the laboratory in the test tube, 20 years later, you should go to the bed. Okay. In between, okay, in between the animal experimentation, safety, efficacy must be good there. Efficacy, optical doses, side effect, another is side effect. All medicine has side effect. Then you have to think very carefully. Either it is, even it is natural products, all it may or may have side effect. So in right side, you see there is some drug discovery pipeline. Okay, drug discovery and development. Okay, so 10,000 compound. Now preclinical studies, this show 250 compound. Clinical development, 5 compound, and regulatory efforts maybe 1 drug. See this here, very more clear. So this is the drug discovery and development timeline. This is the conventional timeline. So this is the drug discovery. So say you have 10,000 compounds. Okay, 10,000 compound you have tested in the laboratory. In the laboratory. Yes, madam. No need to take picture photograph. Just this one. If you need, I can give my slide. Don't worry. Focus. Try to understand. So this 10,000 compound. So you are testing it. First, you have to check things. Either it's active or not active. If active, toxic or non-toxic. If out of 10,000 compound, we are giving say 50% compound. Okay. 50% compound, 20% compound, it is active. Then this compound should be tested, either it is toxic effect or not toxic effect. If compound is very good active, but it is toxic effect, useless, no meaning. Activity is there, no or minimum toxic effect, this compound goes to the preclinical trials. Then these clinical trials, it is in animal. Then again, people do safety, efficacy, pharmacokinetics, pharmacodynamics in animal. Then it goes to human. This clinical trial. Then FDA reviewed, and then the market. So from this one, the preclinical test, it is animal, it is in human. These two part, how it happens normally for the drug discovery, we'll see little by little. Sit here, drug discovery. Okay, this target compound. So we tested many compound. Okay, so we tested 10,000 compounds. Out of 10,000 compounds, some compounds are active. We called it candidate compound or hit. Okay, this compound again we'll test. Then some of the compound will be the safety exhibition. We call it initially the all are compounds. Then those are good activity, good no side effect. We call it hit compound or candidate compound. Out of those compound, those are compound shows safety, efficacy. We call it lead compound. Then this lead compound, from this lead compound, people do a clinical trial in human. Okay, is in human, animal efficacy in animal human. See here, this is basic research in the laboratory, and here is the clinical research. Here we call it translation research. This translation research from test tube to human. In between, there is a place, translation research. Okay, this animal is used here, translation research here. So, if you consider here, the drug discovery procedure here, drug discovery, this one. Let's see little by little. So, initially, all the test is done or run in test tube. If any test is done in test tube, we called it in vitro. In vitro means glass. Okay, there are many in vitro tests. activity, DPPS assay, anti-glycation assay, different MTT assay, this kind of all many tests, the 100,000 of tests is done in either in test tube or 96 cell microarray plate. This kind of test, we call it in vitro test. And what is the in vivo test? It is the test, same test in vitro, in test tube, same test if you do in the living condition, mostly done in the animal, we call it in vivo. Then, in vivo example is animal toxicity, acute toxicity, chronic toxicity, then preclinical trials, clinical trials. This is all in vivo test. So in clinical, preclinical study, it is preclinical. Name is one is clinical. Clinical is human study. Pre means before that. It is done in the animal. So mostly, first we have to see either it is toxic or not toxic. Toxicity. Okay. Then these clinical tests, sometimes we do in vivo animal models, to understand the efficacy, toxicity, and pharmacokinetics. Now, here you are doing some basic research. Here are human. Again, you see. Article is there. Skin goes. Where is the skin? Okay. Yes, online student. Can you see my slide now? Can you see my slide now? Yes. Okay. So the test done in the laboratory and in the hospital or clinical trial, this gap, this breeze, the animal study, it makes the breeze between laboratory to clinic. I found a recent article, how to make some, we call it translation gap between the basic science, clinical trials, and patient care with Alzheimer's disease. So I found a very recent article here, you see it here. Initially, it is done in the drug, then animal, then human, then many human, then ultimately it come to the market. So animal models of disease states. Test condition involving the disease or injury similar to human condition. You want to make normally similar to human condition. So in case of must equivalent in mechanism and predict the human toxicity, at least normally the animal study, the toxicity, it not mimic 100%, at least 70% it can mimic. 70% is enough. It mimics the 70% condition. So how to do the toxicity studies? There are many ways. Okay, first thing is the behavioral study. So if you're taking a good food, your good mood. If you good, same is true for the animal. So you want to do some experiment with your respected drug or your target drug. You are giving this drug or plant extracts to the animal. So if animal don't behave normally, okay, the first thing is the behavioral study. If you're good mood, good food, everything is good. If you're is not good mood, it means you're taking some food. It's not as per your expectation. Same is to pass the behavioral study. Okay, then we do acute toxicity, chronic toxicity in many ways. And one is in vitro test, another is in vivo test, another called is ex vivo test. Ex vivo. What is the meaning of ex vivo? Anyone tell me? Outside the body. Okay, you know, in a human, normally sometimes what happens? Yes, human. Normally, sometimes if you have some kidney problem, we call it kidney transplantation, right? Or sometimes is some kidney transplantation. So how kidney transplantation done? Someone is giving the kidney and someone is receiving the kidney. So if someone giving the kidney, they have to just cut it from his or her body. So this one person to another person, it takes few hours, right? So in these few hours, kidney is functional or not functional? It's alive. It's outside the body, but it's alive. If it is not functional, not alive, the transplantation is useless. Same is true if you collect some organ from the animal, it's alive as it is inside. But the human body, inside the body, there are millions of factors. But if you bring it outside, it is many factors is affected. Okay, because inside the environment and laboratory environment is completely different. Okay, so if you take some organ from the human, from the either it is animal or human, inside to outside, if you do some experiment, we call it ex vivo. Clear? And this ex vivo experiment, what is the advantage of ex vivo experiment? So from single organ, so with short period of time, you can test compared to in vivo many compounds, many things in short period of time.
Should be exactly organ or, for example, biopsy or something like this?
So what we do, I'm giving one example, say ex vivo, say normally in the rat, okay, rat, there is organ, we call it pancreas. In pancreas, the pancreas has islets. The pancreas has normally the diabetes due to less of insulin. The factory of insulin is pancreas. Okay, in the pancreas, we call it islets of Langerhans. This islets contains alpha cell and beta cell. So in beta cell produce insulin, right? So if we collect a pancreas from mice, and if we do some experiment in pancreas, it called ex vivo. Or we collect pancreas from animal, from pancreas. So we purified the islets that contains the beta cell. If the islets, we do some compound, adding some compound, it enhance insulin secretion activity or not. If we do this kind of experiment, this is the ex vivo. So this is beauty. What is the ex vivo beauty? So in vivo and in vitro, one is glass test tube, another is the living condition. So ex vivo, it's mimic the in vivo condition. It is very physiological as in human in living condition. This is the beauty of ex vivo experiment. Yes.
So primary cell culture or ex vivo? Primary cell culture.
This is cell culture. Is you are working on
Cell line, but in primary you get affect the cells from the body.
in the cell line. This is in vitro cell line, but primary cell culture at this moment I'm not sure. I'll check it. But the cell line you normally you're using, it is in vitro. The primary cell culture I'm not sure. I'll check it, madam. Yes, sir.
The our gland was tissue, was the organ part. We make a thyroid solution of that we basically provide the environment, the isotonic solution that body and we that the regional and can perform the agonistic and antagonistic activity in power gland. There is a two examples of activity.
Cell lines are in vitro.
Yeah.
Tumor or something like this. Tumor. No cancer issues in the body. When we extract, for example, the cancer tissues in the body, people or something.
Can you tell me once more? Please, yes.
Cancer tissues like tumor, body, and we try to bring it out, ex vivo.
Ex vivo. It's ex vivo. Any organ, it's any organ. If you do experiment on any organ, it is ex vivo. Then once the animal study done, then we move to the clinical trials. Clinical trials means it's experiment on clinic or human. Okay. So mostly there is a clinical trials. It is the most time consuming and most expensive. Okay, the clinical trials. So initially, drug, to be a drug, its efficacy is important, but safety is more important. Safety is more important. Therefore, initially in the clinical trial, we call it phase 1, phase 2, phase 3 clinical trial. In phase 1 clinical trial, it is done on volunteer, healthy subjects, healthy volunteers, to see their toxicity first. And what is the toxicity and what is the maximum doses it can tolerate. Okay, this is called phase 1 clinical trial. It is done normal, healthy subject, volunteer subject to check the toxicity level and what is the maximum dose it can take it. And phase 2, we call it control clinical evaluation. It is done on patients. And we call it to check the safety, efficacy, and drug toxicity and drug interaction. And this is called single blind technique. That means you are giving medicine to someone and someone is taking medicine. Out of these two, so I am giving the medicine. So if you consider it is capsule, okay, inside the capsule, in some capsule, the real drug is there. Inside the capsule, so it's some fake drug or some nothing is there. But outside looking same, but inside is different. So if I am giving this kind of drug to someone, I know who is capsule contains drug or no drug. So sometimes we call it placebo. Okay. So if I know what is inside, you don't know then we call single blind experiment. Okay, this is single blind. And phase 3, normally it's called multi-center. If it is in case of phase 1, it is 300 patients. If it is multi-center, if it is 2 center, 600. 10 centers, 3,000. It's number game. If multi-center, number of patients is automatically increasing. Sometimes multi-center, yes.
Based on FDA,
Yes. Yes. And phase 3, okay. Phase 3 is do the same. What is the effect of the phase 2? Same thing is done in the multi-center. So, as I told you, so our is normally the ecogenetics. So our genetics is the same, but due to the environment, is many things differ. So if you have 5 brothers and sisters, so if your mother cook chicken or fish, someone like head, someone like leg, you're from the same father and mother, but So liking and disliking is different. Someone like biology, someone like chemistry from the same parents. So patient are different. Okay, so therefore it should be tested on multiple centers to make it more widely acceptable. And number 4 clinical trial is preclinical, phase 1, phase 2, and phase 3. After this phase 3, it goes to the market. So after coming the market, because the drawback of the clinical trial is, so medicine is the same, patients are different. Okay, in the all the world, millions and millions, billions and billion people. But the test is done only some very specific area or a specific country. So as I told you, their physiology and your physiology, it's not same, but similar. Sometimes I told you, a small difference make a big changes. So same drug, okay, same drug may be effective for them, but it may save, but it may or not effective, or it's not effective, it's not a problem. But problem is if it is has a severe side effect and toxic. Same mango in your country, if you take the seeds from the mango and so on, put in other places or other city or other places, maybe tree is okay, but the mango is okay, but taste will be different. So due to the many environment, due to soil, many things. The same is true for human. So we are grown up in different weather. You are grown up in different food culture. You are different in different culture. So some cultural variation, food variation, geographical variation, it makes your human physiology or some many system is different. So after any drug, phase 1, phase 2, phase 3, if the drug came in the market, so sometimes somewhere in some group of people, they find, okay, they're severe, by taking their medicine, they're severely some problem, something toxic. So this drug was withdrawn from the market. This is called post-marketing. We call phase 4. And so as I told you, so it takes 20 years, almost 16 years from bench to market. Again, 40% drug withdrawn from the market, 40%. Phase 4. So then normally only the drugs we're taking normally tested in pharmacokinetics and pharmacodynamics. Once you take the drug, what is the effect of this? How your body react with the drug? How your body react with the drug? We call it pharmacokinetics. And if your drug, after taking the drug, what happens? The drug will go somewhere to active to make it function. So what drug do our body? This is called pharmacodynamics. So after lunch, we'll continue this pharmacokinetics and pharmacodynamics. What is the difference? How it works? Because all the drugs to bench to bed, it shows it must be tested on their pharmacokinetics as well as pharmacodynamics. Okay. Pharmacokinetics is the with time their drug concentration, and pharmacokinetics with time their efficacy. Okay, so after lunch break, we'll see how the pharmacokinetics and pharmacodynamics works, the facts and figures and the physiology, pathophysiology. So with these few words, so I'm taking some questions from the online participants. Anyone send me the attendance link? Okay. So online students, those are attend in online, please give me feedback. So can you hear me clearly? Thus my lecture is understandable. Can you hear me? Can you understand my lectures? Yes, sir. Yes, sir. Yes, sir. Yes, sir. Yes, sir. So Another 5 minutes with you. Yes, we can hear you. Very yes, it is understandable. Okay. If you have any questions, comments, you can write the chat box. Yeah, I'll try to explain or give my feedback as soon as possible. Yes, sir. Yes, sir. So if you have no questions, so we are stopping here now. So exactly after 1 hour, we'll again restart. Thank you very much for attending. Thank you very much for joining. Hope you will enjoy tomorrow after this. So tomorrow's lecture and after tomorrow's lectures, this is theoretical mostly, but next I will explain, I'll share my experience, my expertise, what I did. Sir, kindly explain pharmacodynamics. Yes, I will explain. It will take time after lunch. Thank you very much. I'll definitely explain the pharmacodynamics. So informative. Thank you, sir. Thank you. Okay. Thank you very much for 1 hour lunch break. 1 hour lunch break. Yes. Bismillahirrahmanirrahim. Yes, online students, are you here? Can you hear me clearly? Can you? Either you can hear me. Good morning, sir. Yes. Can you hear me clearly? Yes, sir. Yes. Yes, sir, we can hear you. Yes, offline students, can you hear me clearly? Loudly. Can you hear me clearly? Can you hear me clearly? Are you sleepy? Okay. Just before going to the pharmacokinetics and pharmacodynamics, so just 5 minutes. So my previous part, you have any question or you comment, okay, then I can entertain this question, and then some students join here as well as some students join in online. We'll restart. Yes, sir. So this session is useful for you. Useful for you. So what will you do anyway? So you are still in the morning first time I ask a question, either human is animal or not. Okay, so there is, there's a life cycle now. Many people believe, so animal are active at night. Human are active at daytime. So animal are hyperactive at midnight. Okay, so many people believe. So nowadays, COVID or this internet facilities makes many people, okay, they're active at not daytime, active at night time. Okay, hyperactive at midnight. So if so, if one generation, second generation, it may tends towards some unusual behavior. If a long time active at night time and inactive at daytime, your physiology, as I told you, initially it's okay. But as I told you, we have some biological clock, biological system. If your biological system at young age, at the age of young, hot blood, young blood, you don't care anything, but elected. Every reaction has an equal and opposite reaction. It's a matter of time. So as Dr. Iqbal told us, nowadays, initially, our life expectancy is 50 years. Now it is 74 years. In Japan, I was in Japan, there are 100 years. In 100 years, people in Japan, they're active, dynamic. But in rest of the world, if you are living long, living with disease. So it's life, it's not quality life. So from my lectures, it is very clear that animals are active at night. So this is the lesson one. Okay, take these lessons. Either you will be going towards where? Animals are active at night and more active at midnight. Okay, now think. Okay, don't take it otherwise. Pharmacokinetics and pharmacodynamics. Kinetics means it change in every seconds, every milliseconds, every minute, every times. Okay. So in human body, okay, if it's a human body, if it is human, ठीक है ना? ये दोनों साइड की है ये वाला? What is this? Yeah. Okay. On Bengali, we call it Khan. What is Urdu? Okay. So just remember this one. Yeah, in Bengali and Urdu, we call it Khan. Okay. So some drug we take by orally, some drug we take intravenously, some drug we take muscular, some takes in eye drop, and some drop in the ear. So, okay. So this is Khan. This is part of your body. Okay, this is Khan. This is part of your body. So if you give the drug, what reaction was that? What is the effect of Khan? Okay, so remember this, the kinetics means body. What our body do with the drug. This is the kinetics or dynamics. In kinetics, okay, in kinetics, there is no D. D is absent. In dynamics, D is present. D is present here. So D, that means drug. What is the effect of drug to the body? How to remember? Many people I found normally in the Viva somewhere ask the student question, what is the pharmacokinetics and pharmacodynamics? So many students make it confused, make it just interchange. So how to remember it? So Khan, kinetics. D, dynamics. Okay, this study how our body affects the drug, it is the kinetics. And this study how a drug affects our body, it is the dynamic. Okay, and pharmacokinetics. I just another word, pharmacokinetics. In the positive, there is always make a sound tick 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 tick. So it is time. So pharmacokinetics is the changes of drug with time. Kinetics, changes of drug concentration with time. Okay, and if you are taking a drug, either any system, it comes to their body. Okay, so say if you take food, it will first digest, then absorption. Student drug, it goes to their body. First, it will be absorbed somewhere. After absorption, it will distribute everywhere. Okay. Then anything anything outside the body, it's harmful, poisonous. So after sometimes this drug should be goes out from our body. So if anything's come inside, okay, if door is, sometimes there is a shape of the door. After taking food, you'll be heavy. Then sometimes it's very difficult to exit. Okay, sometimes going inside is easy. Okay, but from inside to outside, because a lot of changes is there, difficult. So the drug should be excreted from our body. Therefore, to make the excretion easily, the drug should be metabolized, should be make it fractionated a small part. It is called metabolism. And then after metabolism, the drug will be the excreted from our body, excretion. So all anything came outside our body, it should be excreted from our body. Or real life example, so at your home, you are used to your father, mother, two brothers, sister, three bedroom. If someone from your relatives or someone relatives came, you can give them hot place, food, shelters, 1 day, 2 days, 3 days, 5 days is enough. 7 days, maximum 1 month. After that, the guest normally they go their hometown and this one. This is the normal physiology. If someone want to stay, what happens? There's conflict. Conflict in the family, conflict. This happens in real life. What happens in a real life to our society? Same thing happened to our body. So this drug kinetics, or we call it pharmacokinetics. It's normally called sometimes called ADME. Absorption, D, distribution, M, metabolism, and E for excretion. This is called pharmacokinetics parameter. Okay, so if you are taking a blood, you are taking a drug from here, this is the medicine. Okay, you are taking orally, so it will go inside. Okay, it will go inside, then it goes to the, it will absorb in the small intestine. If it goes to the small intestine, it will absorb there. Then it will distribute it throughout the body, tissue, cell. Then the drug has some specific action to a specific site. It go there because many people are coming from outside to comstech. Some workshop is here, some meeting is there, some other festival is there. So everybody knows where I have to go. Okay. So after those, same is true. It is absorption first in the small intestine, then it is distributed throughout the body. So sometimes in single system distribution, single system. So where is the blood flow is higher? Say now, the here blood flow is faster. But blood goes to this in our circulatory system. Then from there it goes everywhere. Option 1. Option 2, it may don't go to the first flow of blood flow, it goes everywhere. There is 2 ways of blood of the drug distribution. It is in blood. Okay. It is in blood. Then blood is extracellular, intercellular, extracellular. Then the drug should be goes to the intercellular. From extracellular, it moves to the cellular part. Okay. So once drug in the cellular part, it action started. The cellular part. Okay. Action started. Okay. Then If it is action, this if is distributed everywhere, everywhere, okay? If it is action started, after the action is done, the drug should be excreted. So drug should be excreted from the body. Therefore, so many drugs can be excreted directly, many drugs can't. So then the drugs goes to the metabolism. Metabolism. The metabolism means it's a breakdown of the drug. In the main objectives of metabolism to make the drug insoluble to soluble. If it is insoluble to soluble, it can easily escape from our body. Okay. So this metabolism takes place in where metabolism takes place? Liver. What is the name of the liver cell? Hepatocytes. Okay. In the liver, the metabolism is done in the liver. Liver, it's hepatocytes. You know, if this is the structure of cell, okay, this is the structure of nucleus. In the nucleus, nuclear membrane, just here, this is the endoplasmic reticulum. Okay, endoplasmic reticulum is connected to the nuclear membrane. So in the endoplasmic reticulum, if there is some ribosome, we call it ribosome is there, we call it RAF endoplasmic reticulum. Okay, in RAF endoplasmic reticulum, protein synthesis happens. But if in the lower part here, it is if there is no ribosome, we call it smooth endoplasmic reticulum. So if in rope, if nothing is there, very smooth. If something is there, it will be not smooth. Therefore, one is smooth. If no ribosome, it's smooth. If there is ribosome, yeah. So this metabolism takes place here in a smooth endoplasmic reticulum. Liver, hepatocytes, smooth endoplasmic reticulum of its metabolism take place here. Therefore, sometimes we call it metabolism means all drugs are toxic. Some side effects. It makes detoxification. Sometimes we normally call the drug detoxification takes place in the liver. Then the main thing, so it takes absorption, then distributed everywhere, then metabolism. So as I told you, the main purpose of metabolism is to make the drug insoluble to soluble. Insoluble to soluble. So after metabolism, so metabolism happens in vitro or in vivo? In vivo, in living condition. Normally, in living conditions, sometimes we call it bio. Bio means biology. Bio means life. Okay. So sometimes metabolism also called biotransformation. Transformation drug from A to from B. Though it is a human body, in living condition, we call it biotransformation. So either it is metabolism or it is biotransformation. The objective is to make it insoluble to soluble. Okay, then sit here. So drugs, you're taking drug by orally, it came to the digestive system. Okay, before going to the small intestine, it passed through liver. Okay, it passed through first liver, then then what we call it liver, then it goes to the intestine system, then from there, then some capillary system, it came again to liver, then it goes to the blood. Before going to the blood, okay, before going to the blood, it passed through the, before distribution comes to the blood. So there is a point. So metabolism take place where? Liver, after the action, right? If the metabolism take place before the action, so I am giving an example. So if you purchase 1 kilo sweets, 1 kilo sweets contains say 20 or 10 sweets, right? You are asking someone to bring it from shops. He bring it, and he by himself, before going delivery to you, he tested 1. That means if out of 10 sweets, 10%. Okay, but it's very good taste. He's taking 2. 20%, 20% loss. So if he took 3, 30% loss. Before reaching to the right place, he tastes, and more the taste, more he tastes, the kilogram of sweet decreasing, number of sweet decreasing. Take it now. Same is true in the liver. It goes to the liver first. From liver, okay. If before going to the intestine, if there is some metabolism, before real metabolism, we call it fast pass metabolism. So it's very dangerous. If a drug has more fast pass metabolism, then you need to make more doses. If someone take it 10 sweets, he tested 5, that means 50% remaining. That means you have to order 2 kilograms. At least if you order 2 kilograms, that 1 kilogram will be tested by him or her, and at least 1 kilogram just twice amount sweet, twice amount of money. Okay, then what is the possible solution? Possible solution is, who is taking the sweets, change him or her, or change the route. Okay, change the route where CCTV is there, so that he or she cannot test it without 100% efficiency. Same is true if some drug has higher first pass metabolism, then you have to either option 1, need the higher dose. Option 2, change the route. Change the route. So if you give intravenously, you can avoid fast pass metabolism. Normally, in case of inhaler, okay, it goes directly to the circulatory system. So absorption, distribution, then drug metabolism. What happens in metabolism? Yes. So as I told you, it make the main purpose is make insoluble to soluble. So to make it insoluble to soluble, we call it polar group. If we add more polar group, it will be more soluble. So there is 2 kinds of reaction that make this metabolism. One is called phase 1 reaction, another called phase 2 reaction. Phase 1, it makes simple modification, hydroxylation add some hydrolysation, phosphorylation, some kind of cyclization, this one. In this way, it makes soluble. So if it is soluble, okay, then either it is soluble or active or inactive. Initially, all drugs are active. After metabolism, drug should be inactive. So after phase 1 reaction, the drug may active or may inactive. If it is inactive, soluble, it can exit from our body, excretion. If it is soluble and active, then this phase 1 reaction goes to phase 2. Phase 2, it makes some conjugation. It attaches some groove, big groove. In this way, it makes soluble as well as inactive. So some drugs, after phase 1 reaction, this soluble, inactive, excrete. Some drug, it is in phase 1 reaction. After phase 1 reaction, it goes to phase 2 reaction. In phase 2 reaction, there is some further modification, conjugations, like cyclization, many things. After that, it makes inactive, and it excretes from the body. So some drug excrete from our body through phase 1 reaction, some on phase 2 reaction. And even some drug is not even soluble. So if the soluble drugs, all drugs goes excrete from our kidney, from our urinary system. If not, if lipid soluble, there's no way. Then with some reaction with bile, it excretes from our body through feces. This is the drug excretion. So now see the kinetics of drugs. You are taking drugs. So in your body, what is the concentration of your drug? Before taking any drug, what is the concentration of drug in your blood? Zero, because you're not taking drug, it is zero. So now you are taking drug. You are taking drug, as I told you, kinetics, tick, 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 tick. With time, concentration will be increasing. So this is the kinetics, pharmacokinetics. So it is the time. Okay. So you will taking the drug here. Okay. Here, amount of drug in the blood. So here, initially, you're taking blood, it goes up, it goes up, certain point, then down. Here, you see, there is a point, we call it maximum point. So here, okay, here, its absorption takes place. From here, why it is not goes in this way? Because it means you are taking some food, you're taking drug. After taking all the drugs, goes up as well as if you raise, everybody not goes in the same time. So at the time, if some drugs goes, some molecules goes there, and already some molecule is excreting from our body. So some are excreting, some are absorbing, some are absorption take place simultaneously, not one by one, simultaneously. So in some point, you'll get the maximum point. The point that drug reached its maximum point, we call it Cmax. Cmax. Okay, this is the Cmax. The point where is the concentration highest, we call it Cmax. At the time it reaches the maximum point, we call it Tmax. This is Cmax, this is Tmax. Okay. So if it is the Cmax, it is the 0, then here this is 50%, where 50% drug concentration, we call it equal Cmax. Okay, this is maximum concentration. The time, we call it Tmax. Half of the concentration, we call it half Cmax. half Cmax. If so, if you make a line here and go here, this is the half T. This is half-life of a drug. Okay, see this graph. So this is the time, this is the concentration. So this is the classical kinetics parameter of the drug. This is in the Cmax. And see here. So this Cmax above some point and below some point, drug works in this area, in this concentration. It is not the highest one. In the highest concentration, above of this, and the highest concentration below certain point, so drug only work in this range. The range the drug works, we call it therapeutic window. So if you give a practical example, if your exam is there 100 mark. So if you got 30 marks, you'll be fail. So 31 to up to 40 fail. 41 to 41 is the pass mark. Then your CGPA or first division, second division will be increased little by little. Up to here too. So you will get maximum 80 marks. So there is chance 100 marks. Okay. So that means if you got the 60 marks, 60 plus minus, if you just make it half, 60 plus minus 30, that means 60 plus 30, 90, 60 minus 30 equal to 30. 30 to 90. 30 to 90, this is the same pass mark. Same is true. The therapeutic window, you are getting some maximum point. So below this point, sum of the point, and above the sum of the point, it is the therapeutic index, where your drug is effective. Below the window, it is not effective. Okay, you are getting 30 marks, you are failed. Okay, same is true. So if it is the subtherapeutic window, it is useless. It will have no effect. And if you take drug, and if it goes higher than the concentration therapeutic window, we called it toxic window. One is the therapeutic window, another is the okay. So if this is the kinetics, so up to here, sit here, if it is there, so after certain stage, your concentration will be the fall to the subtherapeutic windows. This is why you have to take multiple doses. So if this is the kinetics from one drug, one capsule, then if you took another drug in this way, that means someone is falling, first dose is falling, second dose is rising. Okay, so second dose is falling, third dose is rising. Therefore, doctor give you medicine, take the medicine after every 6 hours. Take the medicine every 8 hours. So this is the very crucial things. Okay, very crucial thing, because medicine and timely medicine. See here, so your dose is declining. So before it goes to the subtherapeutic window, you have to long before you have to take the medicine. So many people do mistake here. Many people going here and there. They are taking medicine almost useless if it goes to the subtherapeutic window. Therefore, the lesson 1 from pharmacokinetics: if you take medicine at the during the lunch time, or you are go outside, there is no guarantee. You can come back to your home. You can take the second dose. Carry medicine. Either you can carry your mobile phone. It is not important, but carry medicine is more important. So doctor can give you only medicine and this one, but these kind of things, if you know the pharmacokinetics, same medicine, people are taking medicine, same different patient, same medicine, different patient. Result is different. Why? So many people very careful taking medicine. Some people don't care. Okay, this is therefore right time, right medicine should be taken before going to the subtherapeutic window. So always we have to maintain a therapeutic window to make the dose, make the medicine effective. Then now come to the pharmacodynamics. So you are taking the drug now in your body. So drug do some action to our body. It is pharmacodynamics. What is the effect of drug to our body? If you see it here, this is the pharmacokinetics, is the time versus concentration. Our pharmacodynamics is the concentration effect. You are taking some drug. What is the effect of the drug? So this is the pharmacodynamics. Sometimes we call it efficacy. Okay, you see it here. This is the response. This is the response in y-axis, and x-axis is the dose. Okay, you are taking the dose initially in the green. This is the response, and this is another response. Two different drugs. Okay, sometimes if his response is 100, this is the, if some drug has response 100 and some is the less than 100. Whose drug is more efficacy? So which one has more efficacy? Here, one is blue line, another is red line. Efficacy in this way, in y-axis. Okay, that means it is 100%. Here, less than 100. This drug has more efficacy compared to this drug. Here, sit here, two drugs. One is blue line, A, and red line, it is B. So both of the drug, whose drug has A and B? Whose drug, A or B, has more efficacy? A and B. How many A and how many B? Whose drug has more efficacy? Is online student. Whose drug has more efficacy? A and B. A. Yes. Online student, please. Whose drug? Okay, blue line. Any other? A. B. How many here? A, please raise your hand. How many B? Okay. So all are wrong. Same. Both, yes. Sit here. A and B both give same effect. Then what is the difference? Dose, difference is the dose. So they are giving, so if it is the dose, the green line give the maximum effect at lower dose, and red one give the maximum effect on higher dose. So this is the potency. So potency is the maximum effect, minimum concentration. Okay, efficacy, there is a concentration. Okay, so this is the difference between efficacy and potency. So two different drugs may have two different efficacy. Two different drugs may have same efficacy but different potency. So my question is now efficacy in y-axis and potency in the x-axis. More close the axis, it has more potency. Out of this blue and red, whose drug has more potency? Blue. And whose drug has more efficacy? Same. My question is, out of this drug, whose drug is good? A or B? Yes, all student out of A and B, whose drug is good? Both. No, only one. A. Someone is A and someone is B. Yes, answer is A or B? Answer is A. So both of the drug make their maximum effect, 100% effect. A did the same effect, less concentration. B is doing the same effect, higher concentration. Clear? If you want to the market, okay, if you want to market, same thing is available in 50 rupees and same thing is available in 70 rupees. Who is one good? Same thing. So same effect, but different potency. Okay. Here, sit. So here, there is 4 drugs. One is A, B, C, D. 4 different drug has 4 different efficacy, and 4 different drug has 4 different same potency or different potency. Here effective concentration EC50 is same. So there are many possible. Here you sit here, two drug may have different efficacy, different potency. Same efficacy, different potency. Same efficacy, same potency. But give me one question here. If in that cases, one is high, another is low. If two drug has same potency, but efficacy is different. Efficacy is different. So drug A make different efficacy, different potency. Drug A in 100 concentration, it can make 100% efficacy. Drug B at 50 concentration, it makes 75% efficacy. Out of A and B, whose drug is good? A. In that cases, potency doesn't important is here efficacy. If two drug has either same or different potency doesn't make anything. So I'm giving a real life example. You are giving an exam, 3 hours exam. 3 hours exam, 100 students there. One student is very active, dynamic, intelligent. He make all the exam within 1 hour. He submit the paper. He answered 80 marks. And one student, okay, he took 3 hours. And 3 hours he answered 100 marks. The maximum chance, the student A, he will get 80 marks maximum. Second student will get maximum 100. Okay, but reality, he will get less than 80, he will get less than 100. But the very simple calculation, the who answered 100 marks, he will get more mark than 80%. So in that cases, you are doing so in 1 hour, you will do some partial effect. This is good. Or with full time, full answers, good mark, this is good. The second one. So always potency is not important. Sometimes we have to consider efficacy is more important than the potency. There. Why it is not? Okay, sit here. These two drugs. This is the B, this is A. This A drug has high potency, or B drug has high efficacy, right? It makes almost 100% effective efficacy, but A is the drug B has high efficacy, low potency. Drug A has low efficacy, high potency. Out of this A and B, whose drug is good? B. So potency not always that matter. It is the efficacy. Sit here. Now we're moving. So the green line, if you want to test the effect of a drug, its efficacy as well as toxicity. Efficacy and toxicity. See, this is the green line. This is the efficacy. And red line is the toxic effect. We are doing any dose. What is the activity, efficacy? The same dose. What is the toxicity? Up to here, you see it here. When it reaches maximum point, up to maximum point, no toxic effect. The toxic effect start later. The toxic effect start far later. So here you see it here. If you give some drug, initially, sometimes we call lagging time, it has no effect. Then its activity effect start. So from here, from this place to this place here, where the maximum activity done and toxicity just started. We call it therapeutic effect with the toxic effect. You see it here in this bar, no effect. Okay, this is called therapeutic effect with the toxic effect. So after that, it's called toxic effect of a drug concentration. Here, no effect. This is the therapeutic effect, and this is the windows. So this area, this space, we call it therapeutic window. Therapeutic window. So some drug has some wide therapeutic window. Some drug has moderate therapeutic window. Some drug has minimum therapeutic window. So if the drug has maximum, more wider the therapeutic window, more safe the drug. Lower the therapeutic window, more toxic effect. So some drugs, so penicillin, after the maximum concentration, the toxic effect is far later. That means its therapeutic window is very wide. Sometimes in some drugs, in cancer treatment, they are The therapeutic window, very close. Red line, green line goes side by side. So therefore, you see if you get some treatment for the cancer, your cancer cell is destroyed, but side effect, severe side effect, because this kind of drug has severe toxic effect. It destroys the cell, cancer cell, as well as destroys the other cell. Okay, so it is therapeutic window. So for drug discovery, this pharmacokinetics, pharmacodynamics, therapy window, these kind of things we should have to take consideration. How to delete this one, the yellow part? So all the things this we have to consider to make a human model. So human disease model in animals, non-obese type 2 diabetic model, obese type 2 diabetic model, LPS induced multi-organ response, many kinds of model is there, okay, depending on your what kind of model. If someone want to make it in Alzheimer's disease, Parkinson disease, their model is different. If you want to diabetes, this model is different. Dyslipidemia, atherosclerosis, this different depending on your experience. So now I come to the point, ethical consideration for the use of animal. So one thing is respect life, animal, either it is rat, mouse, doesn't matter. Everybody has right to life. Okay, respect life, and you want to, what is the purpose of an experiment? If it is playing game, it's no, it should be something betterment of the human life, betterment for the society, betterment for the society. This is the, okay. And it should be social benefit. And one thing is minimum the use of number of animals. Okay, there is we call it 3R. 3R. One is replace, means it is if possible. So the animal model, if any other possibilities to get the result, replace animal. Reduce, use minimum number of animal. Then R, define, minimize stress and study animals. So if you handle the animal, as I told you, okay, make them, treat them like as your son, as your siblings, as your daughter, like this. Okay? Love, with love and affection. Don't make it aggressive. Replace, reduce, refine. Okay, so this animal with mice, rat, guinea pig, normally we call it a small laboratory animal. Then what is the large animal? Monkey, donkey, dog. This is called horse. Yes, yes. This is called large. But sometimes small animals, if you do some experiment on a small animal, so a small animal anatomy and human anatomy is a little bit different. If you go to the large animals, the monkey, chimpanzee, their physiology, their anatomy very close to human. Okay, monkey anatomy, chimpanzee anatomy, and pig anatomy is very close to the human. Therefore, there are many diseases. So normally FDA, they recommend you have to do toxicity test.
So difficult to compare this treatment amount because they have completed from the system and they have much more since than before that. And my source of similarities on the chimpanzee or monkey. I think ethical must be strict on using these models.
Yes, it is. You can use it, but the problem is that normally to house the mouse and rat much easier, cost effective, environment friendly, everything. But to, as I told you, you have to, once you have some animal model, you have to take care of this. Okay, so to care of small animal is very easier compared to this large animal. Therefore, all over the world normally, so there is two things. One is get some idea first. Anything we have idea, normally we use in a small animal. So even it is FDA, the toxicity test should be done two different species. One is a small animal, another is large animal. So you cannot go directly to the large animal. First, if take some idea from the small animal. So large animal, this is the non-human primate, horse, bovine, sheep, goats, dog, cats. Their anatomy is very close to the human anatomy. Okay, especially the atherosclerosis model, especially cardiovascular disease model, it is normally done in large animal. Okay, large animal, because due to the anatomical difference. So even we are using mice and rats, so that some difference is there. Human, okay, normally what digestive system, human digestive system is physically large. There in the rat digestive system is large compared to the rat. Here, one is anatomy, another is physiology, another is the metabolic rate. Metabolic rate is different from mice. Larger the animal. If elephant, the heart rate of a mice, it is an elephant, which animal has higher heart rate, heartbeat. Mice. Okay. So many things should be considered. So maybe later we can check it. In human, 130 kilojoule kg by body weight. This is the metabolic rate. Mouse, 961 kg metabolic. That means almost more than 5 times. Okay, so these things should be considered when they convert the dose from mice to human. Okay, though their metabolic rate is 5 times higher in mice, 5 or 6 times, the dose in mice and dose in human should be 6 times less. Sometimes there is no direct conversion from in vitro to in vivo and in vivo to human. These things should be considered. We'll do that. So finally, so metabolic disease, right? Metabolic disease, you know, the metabolic disease. So, you know, there is a metabolism everywhere. Taking food, anything we're taking, in our body, metabolism is taking place. Metabolism is nothing; it's catabolism and anabolism. So mostly by metabolism, we think, okay, in general sense, in metabolism, it's breakdown of the molecules. So our human disease, so normally many disease, either the enzyme is working or enzyme is not working, or it is, therefore, many disease, if you go out to the hospital, they give some test. Okay, if enzyme is high or enzyme is low, by seeing the enzyme, we can some predict. So all this enzyme is normally have substrate to product. So if the metabolism, but due to some problem of metabolism, some disease happen, we call it metabolic disease. So cardiovascular disease, type 1 diabetes, type 2 diabetes, there's many dyslipidemia, all obesity, all are metabolic disease. So abnormal chemical reaction. Okay, so out of this metabolic disease, many disease. So though you are working on diabetes, so from next 2 days, I will discuss about you what is diabetes, what is the pathophysiology of diabetes, what is type 1, what is the pathophysiology of diabetes, and if you know the pathophysiology of diabetes, the same things how we can perform, we can make in animal. If we can make what is the things happen in human, same thing if we can introduce in the animal, it is animal diabetes model. If we make it in mice diabetes model. If we make it rat diabetes model. Finally, as I told you, remember why we need this kind of study. Patients are different, but medicine is the same. So same medicine, it may work, someone may not work with you. Initially, diabetes, it is believed, initially it is called Reisman disease. only rich people, the obese people, they develop diabetes. But it is not true at all. So not true at all. So in many senses, the diabetes is different from the Western countries. There is a type 1 diabetes and there is type 2 diabetes. Type 1 diabetes, without insulin, there is no insulin. For survival, they need insulin. But type 2 diabetes may be less insulin or more insulin. If it is more insulin but not active, we call it insulin resistance. Okay. In classical system of diabetes classification, we call type 1 and type 2. If you have some blood glucose level more than 20 millimole per liter, it is classified in type 1 diabetes. But in our experience, we found many patients in Bangladesh, normally it is written that type 2 diabetes developed at 40 plus. But we found many patients at young age, 22, 25, 27, under 30. So their blood glucose level is more than 20. But in classical pathway, more than 20, it is type 1. Type 1 diabetes need insulin. But we found it is more than 27. So by classification, it is type 1. But without insulin, type 1 diabetes cannot survive. But without insulin, they are living. They are not type 1, not type 2. But the problem is this patient treatment being handled or treatment by the classical pathway. It may or may not be correct. Sometimes it may be wrong. Therefore, we have to think a little bit. Okay, this is the figure of Professor Ataur Rahman. Okay, very famous person in Pakistan. Okay, he told chemistry needs a new formula for success. What is the new formula? You see it here. So all the peoples, nowadays what we are doing, we are engaged three things: doing research, publish the paper, and then present the paper in the scientific meeting, and then participate workshop, this one. We are in the circle. Okay, in this circle, by doing this circle, it is impossible to develop drug. Okay, then it is the time to think. If we think, we have to develop our medicine by us. Our pathology is different. So it is the young people, fresh people, fresh mind, fresh thinking, fresh blood. The young people, it is your time. Think and think. Publish paper, present paper, participate. This is enough. Or you have to focus. Another thing, you have to focus to target things focus on the basis you have to do, so that if you start today, as I told you, it takes a long time. If you start today, it doesn't mandatory that you will discover something, but it's the initiation. Someone will run it. So it is the time. So either you'll be in this circle or out of this circle. Okay, so up to now, I told many things about the advantage of the animal model. Everything has some limitations. Okay, they have animal model also limitations. Any models cannot mimic all of the characteristics, clinical features of human, number one. And some differences, physical difference, some anatomical difference, it makes some limitation. Therefore, this difference makes in vitro result, in vitro is good, may or may not be true for the in vivo. If all the things are going well, it fit match very well in vitro to in vivo. Then in vivo animal to human, sometimes match, sometimes doesn't match. Why it doesn't match? Because anatomy is different. Many things is different. Anatomy is different. Even it comes to the market, from market it withdraw. Why? Patients are different. Patients are different. Okay, so this is the things for consideration. The conclusion is bridging the gap, how we can make the translational research. Finally, okay. So I'm from Bangladesh. It is written in Bengali poetry. Shadhinatar Shuk, the happiness of independence. Okay, so Shadhinatar Shuk, some poet write this poetry. There is two bars. So read in Bangla. বাবুই পাখি ডাকিয়া বলিছে চড়ই, কুড়ি ঘরে থাকে শিল্পর বড়ই, আমি থাকি মহাশিকে অট্টালিকা পরে, তুমি কত কষ্ট পাও রোদ বৃষ্টি ঝরে। বাবুই হাসিয়া কয় কষ্ট কি তাই, বাবুই হাসিয়া কহে সন্দেহ কি তাই, কষ্ট পাই তবু থাকি নিজের বাসায়, পাকা হোক কাঁচা হোক পরেরও বাসা, নিজ হাতে গড়া মোর, নিজ হাতে গড়া মোর খাঁচা ঘর খাসা। It means these two birds, okay, so he's living in the palace, he's living in his own house. He's criticized, okay, your house is so small, it's windy, if its brain is there, this will be destroyed, this one. Okay, then he replied, yes, I know this, but you are inside the palace, there is food is good, everything is, but you have no freedom. It is my own house. It is made in either it is no cement, no very good quality, good architecture, infrastructure, but I made it by myself. I feel proud to stay here. It's my own. Okay, anytime, because they are taking you to make you happy. So at certain stage, if something is needed, they'll first sacrifice you. Okay, so this is the difference, because all the medicine nowadays we're taking from somewhere. We have to at least, this is the time to take it. We have to make our own medicine to our own way, our own system. Because in Islamic history, you see, Islamic history, initially, the alchemy, there is lots of history. We have a very historical background, chemistry and medicine. At the time, nowadays, scientists are trying to do some medicine, regenerative medicine, so that you can be young alive. So many years back, the Muslim peoples initiate start, but however, long time it was in sleeping mood or somehow in wrong directions. Now, as I told you, there is cycle, there is cycle, there is circle. Maybe sometimes we'll revive. Sometimes it is the time to think and rethink to how to make it, how to make our own system by using our own knowledge, our traditional knowledge. Okay, in this way, comstech is taking the initiation. I appreciate, and it is our time, it is your time, young generation, to how to make collaboration, how to make cooperation, how to make uplifting the science and technology in scientific world, how to uplifting science and technology by in the Muslim world. Okay, it is your time. It is your time to think, and either you published, present, and participate, or you will do something different. With these few words, I am ending here. Yes, online students, if you have any questions, I can answer first your questions. Thank you, Professor. Interesting sessions. Okay, more yes. This sir kindly share presentation. Any other this presentation? It is my lecture will be recorded, already recorded, and it will be put in the comstech website. You will find it, inshallah. So other than sharing presentation, so tomorrow, hopefully from 10 to 11, 10 to 1, so it will be more interactive. So I'll share my own experience, how I develop mice model, how I develop rat model, how I develop insulin resistance model, how I develop a pre-diabetes model. So there is a lot of difficulties, and how I can, what are the tips and tricks. There is always some difficulties, always some difficulties, always some problems. I'll share my experience, how I make successfully good models, good models. A good model is needed, absolutely needed for your good research. With these few words, thank you very much to attending my class, to attending, and after the lunch, there's your sleeping mood still. Thank you very much for attending the lectures. I think, I hope it will give some idea, some knowledge, how to work with animal, and what is the benefit, what is the limitation of your animal, and what is the, if everywhere is limitation, there is some way to overcome the limitation, And finally, good luck for you. Good luck for you. See you tomorrow at 10 a.m. Thank you. Assalamu alaikum. Thank you.